Yeah.
Right.
A very warm good morning to all. On behalf of organizing committee, myself Amit Kumar Gantani, welcome the eminent speakers and all the participants for national webinar on drug discovery and development using bioinformatics approach, organized by Sani Guruji Vidya Prasarak Mandal's College of Pharmacy, Raga District, Nandurbar. Without wasting much time, I would invite our principal, Dr. S.P. Pawar sir, for introduction of our Pujya Sani Guruji Vidya Prasarak Mandal and our institute. Over to you, sir.
Okay, thank you. Honorable Shri Bapu Sahib Deepak Patil, President Pujyasani Vidya Prasarak Mandal Chahada. Guest of today, Honorable Dr. Yogesh Bacha, Founder Director Apex Pharmaceutical Consultancy Services Mumbai. Second guest, Honorable Dr. Satish G. Polshettiwar, Professor and Head Career Services and Placement Pharmacy, MIT World Pace University School of Pharmacy, Pune, coordinator or PSVP Mandal, Makran Bhai Patil, Shri P.R. Patil Sahib, my fellow collegians, as the principal and the convener of the national webinar on the drug discovery and development using the bioinformatics approach, I welcome you all on behalf of a student, member of teaching faculty, non-teaching faculty, and myself. Dear students, this Pujyasani Guruji Vidya Prasarak Mandal, established in 1969, the Mandal has serving the great cause of education in the remote Adivasi part of Maharashtra. Sir, this area is mainly populated with the economically and social backward poor peoples and the landless Adivasis. Educate the young generation of such a poor society, poor section of society. Mandal started its Arts, Commerce and Science College in 1969. Later on, College of Engineering and Polytechnic in 1983. Then Industrial Training Center in 1985, College of Education 1986, Management and Research Institute in 1987, College of Pharmacy in 1995, Advanced Studies Center for the English and Indian Languages in 1995, College of Agriculture was started in 2003, Junior College of Education was started in 2004, and developed with the all infrastructure and educational facility at Mandal own continuous 250 acres of the land. The Mandal honestly desire to offer quality education to young generation and make it competent, energetic to face the life in all difficulty, natural as well as the man-made. Now let me furnish some information of our College of B Pharmacy. The course B Pharmacy of Pujyasani Guruji Mandal Vidyaprasarak Mandal was started in 1994 under the kind and able leadership of late Honorable Annasap P.K. Patil. Sir, I am also happy to share that the college was accredited by the NBA and got A grade from the North Maharashtra University, Jalgaon. Sir, it is approved by the All India Council for Technical Education and affiliated to North Maharashtra University, Jalgaon. 12 batches of young pharmacists have passed with a flying color. Many of them are serving pharmaceutical industries institute in India as well as abroad. Our college is located in a peaceful and natural surrounding of Pujyasani Guruji Vidya Prasarak Mandal's campus. The college run in the 3-story building with additional basement nearby, accommodating to all necessary facilities like volume library, well-equipped laboratories, classroom, amenities for the sports and extracurricular activity. Students are encouraged for the preparation of various national level tests like GPAT, NBA, GRE, TOEFL, as well as placement has been started in our college to provide the job opportunity to our students to campus interview. Last year, our college conducted the campus interview and 89 students from all the campus were given the jobs. Even every academic year, sir, I'm happy to share that in every academic year, one national level seminar conducted and successfully organized by our college that provide an opportunity for our student and to interact the academicians, industry person, well-known personality from pharma sectors in inviting as a speaker every year. Also, one day workshop for the personality development for the girls is successfully organized by our college under the heading Yuthi Sabha, sponsored by the Student Development Section, North Maharashtra University, Jalgaon. I'm very glad, proud to say that Phase 1 Nandurbar district level Avishkar, university level Avishkar was successfully organized by our college for the efforts taken for successful organization of this event. Our college was appreciated by the university authority. We also highlight and aware the local public for the social issues like cancer, AIDS, Swachhata Abhiyan, attack, etc. by organizing the rallies and street act every year. We have adequate and number of the qualified staff members, competent and energetic. Their efforts in molding our student into the well-informed, self-disciplined professional is appreciated. They always motivate the students, so our students also should qualify the competitive exams like ZPAT. For this purpose, regularly organize the guest lecture. Along with the curriculum, various extracurricular activities also conducted to encourage the students, like organizing the Freshers Welcome Day to remove the language barrier and to make interactive. Our teaching staff members are also doing sincerely. They are cooperating in conducting the extra classes for the various subjects as per need. They are also involved in the quality research work publication, the work published in the national as well as the international journals. Lastly, I expect the graph of success to move ahead in the future. This webinar will be helpful to upgrade the knowledge of young mind towards new and emerging field and will surely help in making the life better than today. I would like to thank the speakers for giving their valuable time for us. Like to congratulate the organizing committee and coordinator for successful organizing of the webinar. I am also thankful to our beloved President, Honorable Bapusab Deepak Bhai Patil, and Honorable Coordinator Makarand Bhai Patil for always guiding us and motivating us to do the best. Thank you very much.
Thank you, sir. Now I would like to invite Dr. Sunila Patil Madam for introduction of guest.
A very warm good morning to all of you. On behalf of Organizing Committee, I, Dr. Sunila Patil, heartily welcome all delegates in one-day national webinar on drug discovery and development using bioinformatic approach. This webinar will focus on new approaches to discovering the drug. My great pleasure to welcome our research person of this webinar, Dr. Yogeshwar Bachcha sir and Dr. Satish Bachcha. Mr. Yogeshwar Bachcha is the founder and director of Advanced Pharmaceutical Consultancy Services Mumbai, and Dr. Satish is associate professor and head career services placement at Dr. Vishwanath MIT WPU School of Pharmacy, Pune. So we shall now start the first session of this webinar by Dr. Satish Bachcha sir. I hereby request all the participants that their questions or queries are most welcome. You need to send their questions in live chat box so that they can be discussed during the question answer session. I feel privileged to introduce the honor of our resource person, Dr. Satish Bachcha sir. A huge in career of science, but some of our activities I want to highlight here, such as some have qualified the international exam. He is a qualified student, 99% from IIT Madras. He is having his own YouTube channel with more than 13,000 followers. Also, he is awarded with various mind-blowing awards like Best Faculty in History, Best Institute, Recently awarded 2019, also with Albert Nelson Lifetime Achievement Award 2018. One more thing which I want to highlight here. So he was nominated for prestigious Beauty Award in 2017. Sir has published more than 45 research and review articles in different national and international journals, along with review of claims, and also he is having 3 Indian patents on his name. Sir has also received grants of more than 20 lakh rupees from Pune University and AICTE. One is a dynamic personality who is well-known speaker for many national and international webinars, conferences, and EFDP programs. Now I request Dr. Satish Shettiar sir, please take the charge of session.
Yeah, thank you. Thank you, madam, for such a nice introduction. I'm audible to you?
Yeah.
Yeah, let me share my screen, then I will start the session. Yeah.
मेरा बंद कर दो. Mic.
Yes, sir. Distributes. Yeah. Let me know. Yeah, I'm visible and audible.
Yes, sir. You are audible.
Sir, can you see my presentation? Is it visible?
Yes, sir.
Then we'll start.
Okay.
Yeah, thank you very much. At the outset, I would like to express my sincere gratitude to the managements of PSGVV Mandal's College of Pharmacy, Shahada, and I express my sincere gratitude to the very dynamic and very fascinating principal, Dr. Sunil Pawar sir, who is taking lead and taking ahead these institutes at remarkable level. So I would like to express my sincere thanks to the today's coordinator, Dr. Sunila Pawar ma'am, for taking this initiative and coordinating these events in very nutshell manner. Also, I would like to thanks to all the participants who have been joined for today's very fascinating and very important session of today's need of hour, we could say, that is bioinformatics approach. So today's topic of my discussion is nothing but the repurposing of antibiotics and biosimilar by using pharmacophore and bioinformatics approach. It's a kind of revolution in research. All of you know the world is facing the problems. Now nobody has thought about today's day that you have been completely locked down since last 3 months at your home. Definitely, it is a challenging environment to one and all. We have to think about that how we can cope up with the challenges. So this topic would be really, give the insight on how we can move ahead and how we can repurpose the existing drug which are there already in the markets in order to cope up with the challenges, in order to reduce the cost and time and pace of drug delivery process. And biosimilar market would be the next future market for the pharmaceutical industry. So one has to understood our student and research scholar must concentrate on this kind of research. Definitely, we have the computational tools. Bioinformatics approach. This approach would be useful for making our drug discover at a very simpler manner. So let us continue with our today's topic of discussion. I hope I'm audible to you and visible to one and all. Let us continue with this. Yeah, yeah. Before opening my session, let me open my disclaimer. Just a minute. Yeah. Today's presentation, what are the content that of the slides are intended for educational purpose only and purely for personal use. And next, the presentation is based on publicly available information that is we call as OER, Open Educational Resource Material. And the views presented are the views of presenter, that is myself, not necessarily to author, employers, organizations. So this is my disclaimer. Let us begin our topic today. What I'm going to highlight today, all of you know, the world is facing the problems. So the growing interest driving towards the computational modeling, like we can say the virtual screening and all in silico library and kind of virtual things are going. Today also we are connected through virtual platform. So thanks to these ICT tools, because of that we could connect each other. Though we are talking about the different potential research after post-COVID, because the world is going to be changed after post-COVID. We should ready for that. We have to make ourselves future ready. That is the need of our. Then we have to understood the antibiotic resistance. That is one of the global threats to mankind that I'm going to talk. Then lab to clinic translates and clinic to lab. That is the research, how it has been translated and transformed from the clinic to lab. That is the need of our. One has to get the many answer for that. Then I'll be talking on a pharmacophore approach. That means finding out the best moiety, best molecule to get the biological activity. That is called as pharmacophore approach that I'm going to talk. Definitely, I will take you on the virtual tour. That is the one fascinating tools of fascinating topic of today's discussion. So how we can get the virtual screening, how we can get it done, all the in silico library utilized for the drug discovery purpose. Next topic of interest is accelerate the drug development through repurposing and repositioning the existing antibiotics. We all of you know since last 10 years, very few antibiotics appear in the markets. So that I'm going to focus on this, how we can repurpose the existing antibiotic and repositioning the existing drugs for the new indication, new clinical indication. Then next topic would be the bioinformatics database. That is today's topic, you all of you know. The bioinformatics approach, computational modeling, how we can use this database for retrieve and getting the your answer solved. That is called as computational modeling through high throughput post screening and high content screening, that is STS and HCS. Then biosimilar market, genomic medicine, that is the future medicines, how it has to be developed and what would be the strategy of any scientist for going forward toward biosimilar. The next topic of my interest would be CADD, that you all of you know PCI recently introduced this computed drug delivery system and drug design in MPharm's curriculum that will facilitate the rational drug design and developments. That is really need of our. Then how to engage our students and research scholars during this pandemic, that everybody is facing the problem, how we can engage our MPharm students, especially during this pandemic situation. Definitely this talk, this entire 1 hour talk will be, give the insight how we can make our strategy, how we can move further for the research and developments. So our former president, Dr. Abdul Kalam, rightly says, okay, in order to, empower the Indian population, one has to understood the IT. Means you have to integrate the IT, that is the industry, we call it information technology, with Indian talent, that is IT. So IT plus IT, then we can make India's tomorrow. That is, we can make India's tomorrow a better tomorrow. That is the statement made by our former president. So that is we call as Industry 4.0. So all we are using the, all of you know, we must understood this ABCD, artificial intelligence, artificial blockchain technology, then artificial wisdom, then we have the cloud-based system, then big database, all are nothing but the part of ABCD. So A is nothing but the AI technology, that is augmented reality, artificial intelligence. Nowadays it is widely accepted and widely be used in pharmaceutical industry. The next would be the B, that is the blockchain technology. We all understood in they are using blockchain technology. Even pharmaceutical industry, for the supply chain management, they use this AI technology and blockchain technology. Because of that, we could connect each other. They can get all this query being solved very instantly. Then C is cloud-based. Everybody is believe on cloud-based system. This was introduced by Amazon 2006. So cloud-based, all of you know, in mobile phone also we have cloud. So we believe on cloud server. So maybe the next future would be on cloud-based system. Then D, your last is D, ABCD. So D is nothing but data science, database. We have huge database. Only we have to apply proper algorithm in order to get your queries solved. That is computational modeling. Because of that, we could connect. You get all information very in a single click. There are a number of software database are there. There are about 1.5 billions. That proteins have been identified up till now. So only thing is that you have to utilize all the proteins for the development of your ligands, maybe by different approach that I'm going to talk in today's session. So we are moving from penful to penless. All of you know now the drug delivery and all coming in penless drug delivery. That is all of you know. Okay. That is shift from classical to modern virtual methods. We are moving from traditional era to modern virtual methods. Okay. These are the four potential fascinating areas of research after post-COVID. Today, I'm going to talk on the third one, repurposing of drug, biosimilar future pharma industry, and the computational modeling, a cloud-based virtual screening, that is IoT, Internet of Things, exploration, that is need of our. There are that coming in future. That is, we are making the technology for artificial wisdom rather than artificial intelligence. So wisdom is important. We have to convert the drug for well-being of society. That is very important. Then telemedicine, all of you know now it is being fascinating. That is because of eye doctor, eye pharmacies coming into picture. That is called as future of telemedicines. Then machine learning, deep learning is the part of component of AI system, that is Industry 4.0. Then wearable biodevices would be the next area, fascinating area after post-COVID, that will give the insight on real-time monitoring of the ADR system, that is called adverse drug reactions. So these are the the areas where we can work and we can involve our student, engage our student to move forward, to get it, make them more and more empowered. That is the very important objective of any institutes. So online outline for future research roadmap. You all of you know today the world is going for omics. So what is this omics? Because we fully understood DNA, RNA. We all of you know the bioinformatics tool is the computational tools that involve the making of the complex structures from the simple one. That is, we understood DNA fully, we understood proteins fully, we understood RNA fully, we understood all entire genomic sequence. Because of that, we could get all this kind of solution. So that is called as omics. Omics means we must understood genomics, proteomics, metabolomics. They are the area coming in future. So because of this all sophisticated technology, like you all of you know, NMR technology, crystallography technology, this because of this technology, we could understood the 3D structure of proteins. So it's become very easy for all the scientists to understood and to to interact your drug molecule with the receptors, and we can possible interaction to get a possible, you can say, the biological activity. That is the need of Earth. That is beyond genomics. There are a number of things are there. So only thing is that we have to, how we can utilize this all database that is called as biological information. That is bioinformatics, biological information. We have the we understood fully all genomic sequence, that we are moving from you all of you know personalized medicines. Now the today's system is fit for all. Whatever tablet, capsule, and all medication you are taking, that is fit for all, without considering the genomic and personalized, disease and all conditions. Maybe the next future would be the personalized medicines. So doctor maybe prescribe the medicine based on a genomic report. Maybe doctor will ask you, send your report on your WhatsApp, on your Gmail, we will prescribe the medicine. That day will come, maybe after 1 or 2 years. Already it has been started in US. If we just talk about the personalized medicine, that is the future. That is called as omic revolution. That is metabolics. All of you know this fluxomics, proteomics, transcriptomics, and genomics. They are the area where the scientists are nowadays working. If we just talk about all these things, because we understood fully all DNA sequence. Next, molecular biology. Okay, need to understood to make a future medicines. For that, one has to understood all the molecular biology, that we are moving from the traditional aspect to the modern virtual method, modern methodology. That is called as. So I'm going to show you one how this molecular structure animations work. Just it's a 2 minutes video. I hope you all are getting. Okay, let us look at the molecular structures. This is one example just to make you understood how does it work. Now you can see the protein molecule, how it interact with your ligand molecules. Look at this video. Okay, we can see the contact points. It's lock and key model. Simply you have to find out the proper ligand that will fit, that is having proper coordination, proper orientation toward your lock. Like you all of you know, you have 100 keys and we have one lock. So you have to find out which particular key is going to fit with your lock. That is, lock is nothing but your receptors, and key is your ligands, that is drug molecules that you're going to discover, that you are going to repurposing. So we fully understood this space fill model, surface model, ribbon model. Because of we could connect, we could understood the 3D structure, entire orientation, coordination of that molecule we understood. Because of that, it is made possible, and it become very easy to get all these things. I hope you understood this. Now we'll move to the next part. You all of you know drug discovery development process. We have been learning since inception, since your B Pharm, M Pharm level, drug discovery development process. Okay, so the participants from faculty, some of the students, some of our research scholars, definitely this talk will be helpful to them. So I'm going to talk on very basic nature, basic things that one has to understood. How this drug discovery development process works. You all of you know, whenever you're going for drug discovery process, what we are thinking, you have to identify, going with the in silico approach and all, you have to first identify disease. Okay, so identified disease can be, you all of you know, the n number of diseases are there. Up till now, if you see, 14,000 diseases have been identified. Okay, that is the recent. Then for that, next you have to identify the protein, because you all of you know, every disease the proteins involved. You all of you know are receptors. Either it would be act as by stimulation or inhibition. We understood fully. Some are inhibitory proteins, some are stimulatory proteins. So for that, we need to identify that proteins. We have to isolate the proteins. We have n number of techniques for isolation of proteins. We have the technique like NMR, we have crystallography technique, X-ray crystallography. Because of that, we could understood 3D structure of the proteins. So it become very easy to relocate, reorientation, all these things. But normally, if you go with this isolation of protein, it takes 2 to 5 years. If you go with normal process, but if you just apply the in silico approach, pharmacophore approach, definitely that would be done within a 1 hour, within 1 week or one year only. So that is the the breakthrough innovation in field of pharmaceutical industry by using bioinformatics approach and computational modeling. That is, nowadays we use molecular modeling words. Then we go to find the drug effective against disease proteins. That is ligand finding. We all of you know the strategy for getting the new ligands. It could be the receptor based, the structure based, yeah, ligand based. We all of you know that process. Either we know the proteins or unknown the proteins. There are multiple aspects are there. I'm going to talk on in my further session. But normally, if you go with this process, it takes near about 2 to 5 years to finding drug effective against disease proteins. Then after disease protein, then we go the preclinical testing on animals. It takes near about 1 to 3 years. We understood fully all the process. Then after that, we go with the scale-up process. Then we think about formulations. So preclinical testing, then we go with the scale-up study and formulation aspects. Then after then we apply file IND. All of you know IND is getting filed through our regulatory agency. We have this CDSU under DCGI. We all of you know in India. But if you go to US, they have different FDA. They have different kind of regulatory agencies out there. But India, we follow we apply to DCGI through CDSU. That is file NDA. Then after getting NDA, then we conduct the human trial, clinical trials that take near about 2 to 10 years. It's a very lengthy and time-consuming process. Then after that, we file NDA, new drug applications. Then after that, you will get approval from FDA, that regulatory agency. It takes 2 to 3 years. Means entire process take near about 20 years, more than 18 to 20 years. It's a very lengthy process if you go with the traditional approach. But if you just apply this in silico approach, pharmacal modeling, this 5-year process can be done within 1 year. That is the beauty of a pharmacal approach, bioinformatics tools. We are really fortunate to have this huge database. There are about 150 database we have been generated up till now. Only thing is that you have to just explore that database for making your drug discovery process very, very simpler form. That is all of you understood. So this is the process how if you go with the traditional techniques, it takes about all of you know 10 to 17 years, and success rate is only 10%. You all of you know, means you're bringing the molecule from this to this. The only 10% success is there, and finally you're getting only one molecule. But if you go with this drug repurposing process, maybe reduce the timing 3 to 8 years instead of 10 to 17 years. That is the beauty of this, drug repurposing, and you all of you know the computational modelings. Directly you can bring the molecule into clinical trials because already the clinical trial have been done by that innovators. Because we are taking the molecule for repurposing, which are being approved in Orange Book. You all of you know, Orange Book we refer those drugs which are being patent expires. That is the para 1, para 2. We understood in regulatory approach, regulatory agency, regulatory guidelines. Okay, got it. So drug repurposing would be the most promising areas for all pharmaceutical industry. Then we'll be talking on antibiotics. All of you know antibiotics because my focus would be on antibiotic and biosimilar products. So it has been said that by 2050, this superbug means nothing but the MDR, that is multiple, all of you know, resistant species. Superbug could kill 10 millions of people in a year. Means where the morbidity and mortality rate is very, very higher, that we refer as superbug. We understood the Pseudomonas species, S. aureus, and number of bacterial species are there which are being become a superbug. So industry are afraid to bring a new molecule and discover new molecule against this kind of superbug, because all of next day they will come to know that drug is not effective against that particular antibiotics, that particular microorganism. Escape we usually consider. So India has one of the world highest level of antibiotic resistance due to the over prescribing by medical professional. Unnecessarily you are taking the medicines. It has been observed that 80% of the medicine which have been prescribed for RTI, respiratory infection, have been unnecessarily taken. Unnecessarily you are giving burden on your system to become a drug molecule, that particular drug molecule become resistant against that species because of over prescription, because of lack of knowledge. Then excessive consumption of over-the-counter version. All of you know, if you just go to any pharmacist shop, easily you will get the medicines, which is being scheduled H drug actually, not to be prescribed, not to be dispensed without prescription. But still it is being prescribing. Still it is being distributed to the common man. So that is the real problem one has to tackle. Then abundance of drug used in chicken and other animal food products. That is another area one has to understood, because the superbug can be generated by different factors. It could be the patient oriented, it could be the drug oriented, it could be the society oriented, it could be the environmental oriented. A number of factors are there. So MDR is among the top 3 global threats. All of you know MDR, multi-drug resistance. Multi-drug resistance is among the top 3 global threat as per the WHO survey. This is the the area where we can target. So if you just talk about the history of this discovery of antibiotic and all that, we understood the penicillin was the first drug, or first antibiotic we could say, which was discovered by Alexander Fleming in 1928. Later on, he got a Nobel Prize in 1945. He he stated, he has made a statement in that time. It is not difficult to make microbe resistance to penicillin in laboratory by exposing them to the concentration not sufficient to kill them, and the same thing has occasionally happened in the body. And by exposing his microbes to non-lethal quantities of drug, make them resistant. Throughout the history, we have been continuous battle between human being and our microorganism. Recently, we have COVID-19. We have continuously battled with microorganism. So study has been always say there are a number of bacteria evolving because of rising temperature. There are a number of factors, climatic conditions. Many microbes are evolving from ice. All of you know, because the melt is getting, your ice is getting melt in African countries, other countries, European countries. Because of that, multiple species have been evolving. Maybe that could be the, one of the threats to mankind. One has to understood that. So AMR, that is antimicrobial resistance, continue to be the public threat on global scale. So how does this antibody resistance develop? All of you know, normally what happens, this is the microorganisms. If you use any kind of medication, antibiotics, normally it has to be kill this particular microorganisms. But what happens over the period of time, what happens, the species become mutant. If you take that drug medication frequently, then that normal species become mutant. What is mutation? Nothing but the change in genomic sequence. We understood a DNA sequence, adenine, guanine, sequence we understood, full AGCT sequence. That sequence getting changed, and that drug will not, again lock and key. That key is not going open to that lock, because initially it was open, but now because of resistance, that key is not going to fit with your lock. That is, all of you know, lock and key model. So because of that mutant species still survive in your body. It has been observed that till 1 year, the microbes will be survive in your system till 1 year. That going to be, produce the resistance effect. All of you know, in case of tuberculosis therapy, in case of, we understood various respiratory infections, resistance develop very, very fast. One has to understood that. This continue to produce offspring that resist antibiotics. So MDR already have told among the top 3 global threats. So this microorganism are playing vital roles. That is called as escape. What is escape? Escape means Enterococcus, then Staphylococcus S means Klebsiella, Acinetobacter, Pseudomonas, and Enterobacter. They are the species that is we commonly call as escape pathogens. That is the responsible species for MDR, that is multi-drug resistant species. That we understood. Okay, if you just talk about this kind of, how resistance develop, it could be because of efflux mechanism, it could be because of lack of proteins, lack of number of factors out there. Let us see this video. We'll understood how this microbes
are tiny organisms that often multiply rapidly. Everyone carries good and bad bacteria. If there are too many bad bacteria, a serious infection can develop. That can make you very ill. The bacteria that cause such infections can be controlled using antibiotics. Antibiotics kill the bacteria or inhibit their growth. This helps your body to recover. Sometimes bacteria are insusceptible to an antibiotic. They are therefore resistant. These resistant bacteria have protected themselves against the antibiotic. Antibiotics kill off susceptible bacteria, giving resistant bacteria the chance to multiply. The infection is therefore caused by resistant bacteria. This infection is more difficult to treat because the usual antibiotics do not work. So doctors have to choose antibiotics of which they have less knowledge or that have more side effects. If antibiotics are used incorrectly or too often, this increases the chances of bacteria becoming resistant. So it's important to carefully consider whether antibiotics really need to be used. This way.
So this is the things, means one has to understood how, where, and we can use the antibiotic. We have to use judiciously and rationally this antibiotic. That is the outcome message from this. Okay. So this is very important. So one has to understood this. So antibiotic resistance could be developed in bacteria. It could be developed in endoparasites like flea and ticks. It could be developed in viruses. Just now we understood resistance to antiviral drugs. A number of drugs have been evolved that is producing resistance. Then fungi, all of you know. Then fungi. Then cancer cells also. Many antibiotics have been used in cancer therapy. So cancer therapy also produces so many antibiotic resistance to antibiotics. The next would be a journey of, if you just talk about the journey from first antibiotic discovery to fully synthetic drug and computational drug delivery. The first drug, we all of you know, penicillin, then streptomycin, then a number of antibiotics have been evolved till 1960. But in 1960, then 1980 was the that period called as lean period, where the maximum resistance have been appear. So 1960 was the golden period till 1960. But later on, the many, because of this resistance, increasing the antibody resistance have been appear. Then if you just talk about 2000, 2020, a discovery getting wide. Why discovery getting wide? Because of this superbug, because of the MDR. That's why the industry are afraid to bring new molecule into markets. The next would be just talk about the discovery and the antibodies. If you see discovery getting wide after 2000. Okay, if you see 1987, 1990, 2000, very few antibiotics appear in markets. Next, mechanism. Just now we talk about mechanism of antibiotics. Okay, how resistance have been developed, because it could be intrinsic, it could be acquired. Mostly, all of you know, acquired is the responsible. 80% cases, acquired is the one of the reason for getting antibiotic resistance. Intrinsic could be lack of targets, lack of the cell wall and all. Innate efflux pumps. Many bacteria cell have the efflux pump. Because of that, drug is coming out. Drug is not getting inside. A drug is coming out. Drug getting that because of PGP inhibitors. So many inhibitors have been there on the cell wall of bacteria. Because of drug inactivations, all of you know, many drugs have been inactivated, like cephalosporin. Because of this, your Klebsiella species drug get inactivated because they release some enzymes. Because of that, your drug get inactivated, your antibody get inactivated and produce the MDR. Then biofilm formation also one of the mechanisms, but mostly acquired. It could be genetic methods, that is because of chromosomal methods which have been observed in all of you know tuberculosis species. You all of you know in microbial tuberculosis species, the therapy we use 2 months, we use this for important antibiotics. We use always in combination therapy. Why we use in combination therapy? Because of this MDR. That is because of chromosomal methods mutation. This is because of lot of insertion, deletion mutations have been happens in your tuberculosis species. Then extra chromosomal methods, it could be because of carrying the genes. Genes get transferred because of the translation, transduction, and you all of you know transformation. The 3 mechanisms we understood in biotechnology, biochemistry. Okay, that is how gene getting transferred from one molecule, one bacteria to another bacteria. Because of that, the bacteria become resistant. This is the mechanisms. Okay, already we discussed that. Okay, I'm not again coming over there. There are different target sites where we have to targets. If you just talk about the microorganism, bacteria, the cell wall, mostly beta lactams, that is vancomycins and all penicillin, ampicillins, target on cell wall. They inhibit the cell wall synthesis. Then some fluoroquinolones and rifampicin, like this antitubercular drug, which act on DNA RNA synthesis. We have to always look at the target sites. That is nothing but the protein of interest. Then and only we can get a ligand of interest on that. Okay, so one has to understood this. Then folate synthesis, some drug, like trimethophan and clotrimazole, sulfonamides, all of you know, which is act on this folate synthesis. They stop the folate synthesis. These are target sites. Then cell membrane, like daptomycin is the one drug antibiotic which will act on cell membrane. Then some drug act on the protein synthesis, some are inactivating enzyme. So they are the antibody resistance. It could be because of efflux mechanism, because of immunity bypass, because of target modification and inactivating enzyme. They are the mechanism of drug resistance. How does this microbes become a super bug? Because of the efflux, you all of you know efflux mechanism. Efflux means coming out. Drug is coming out from the cell. So you all of you know drug, once the drug is coming out of cell, it will not show any biological effect. Then ultimately drug dose would be increased, and because of it produces resistance. So one has to think on that. There are different target sites and resistance antibiotics. In case of tetracycline, the target site would be the ribosomal point of mutation. We know 30S and 50S ribosomes. Then altered DNA fluoroquinolones. In case of penicillin, they target on modified penicillin binding proteins. Then in case of rifampicins, they act on mutation of DNA dependent. They are mostly target site is DNA dependent RNA polymerase. That's why we use in combinations all the therapies. This is the one mechanism how drug getting your antibiotic become a resistance. Now see, initially this is the antibiotic which is having spherical shape. Now it is its binding capacity, affinity is very good initially at normal level. But what happens later over a period of time? Now see the antibiotic structure is remain same, but the receptors is going to be changed. Now see the shape have been changed. Because of that, what happens? Because of this block the binding sites, it will not show the effects. This is a kind of happening inside the cellular level, molecular level. So what is the idea of repurposing now? You all of you know repurposing means nothing but you have to repurpose the existing molecule. Now recent example we know hydroxychloroquine, which have been used for anti-malaria, but now it has been used for prophylaxis of COVID-19 therapy. So recently I heard yesterday only dexamethasone for prophylaxis of the COVID-19. These are the drugs now being coming. Now scientists are using this computational modeling for repurposing of drug. So only thing you have to apply proper strategy. You have to work very judiciously on that, rationally on that, so that we can get a various solution on that. So why there is a need of our to investigate the new use of existing drug, that is called as drug repositioning or drug repurposing. We have to repurpose the existing drug for new indication, a clinical indication, that is called as repurposing. So discovery and developing new drugs need to high expenditure. All of you know the cost involved in more than 500 billions dollar required for discovering new molecules. Even pace is very slow and time consuming process. So prolonged timeline are major roadblock for creating therapies for infectious disease. So drug repurposing is a safe and successful pathway to speed up the novel drug discovery process and development process. So there are now many scientists who have gained the renewed interest in convenient and effective way by repurposing their drugs in order to recognize the global health threats that demands effective solutions. So why we investigate the new existing drug for so mostly we use the FDA approved drug. All of you know once the drug get FDA approved, so it will be appearing in the your all of you know orange books. Okay, so we can refer the orange book for the patent expiration and we can work on that. So that is the first priority. Then drug that fail in clinical trials that can for the further repurposing, which have detailed information on potential toxicity and formulation pharmacology. No need to again do all kind of study because Innovator have done all the kind of clinical trials, clinical study. Only thing you have to think in different perspective. Technical advancement in the informatics, you all of you know informatics, bioinformatics, genome informatics, pharmacotherapy, all are being evolving subject in the pharmaceutical industry. Genomics and biological science account for major success of drug repurposing in identifying secondary indication of existing drug. We have to find out the secondary indication. We understood fully, and number of drugs have been repurposed. Best example, your aspirin, you all of you know, that is being nowadays used for the antiplatelet agent, but it was used for NSAID initially. That is a kind of indication. The new repurposing or drug repurposing is playing a vital role in filling the gap in the discovery of potential antibiotics and all of you know drugs. Bacterial infections are emerged as a ever-increasing global public health threats by dint of multi-drug resistance of existing drugs. This raised the urgent need of development of new and repurposing existing antibiotic that can effectively fight this multi-drug resistant bacterial infection, MDRBIS mostly. What a strategy we can use for that? So we have the virtual library, that is in silico library. We have the software tools. We have database. We have the zinc database. We have the Gomez database. N number of databases are there. There are about 150 databases are the protein database we have, PDB database we have. We have the drug bank. We have the various ligands molecules we have. Only thing we have to just interact that molecule with your protein of interest. So in silico drug repositioning platform represent alternative and complementary approach for activity-based drug discovery. So we can find out the possible interaction, possible best fit model for your ligands. If you know the proteins, then we can just search that proteins, which particular proteins we are going to target. So there are different strategy we can use. Either we go with receptor-based study, go with ligand-based study, a structure-based, that is called as protein-based. So all of protein-based means what happens if you know the proteins, then we can find out the proper ligand for getting fitted with that molecules properly. If you know the both, that is receptor is known and ligand is known, then we can go with the structure-based drug design process. If you know the receptors and if you don't know the ligand, then you can go with de novo methods. We all of you know we make the complex molecule to simpler molecule, but it's de novo methodology. If you don't know the receptors, that is receptor is unknown, then ligand is known, then ligand-based drug discovery process. That is the approach. That is we call as theory behind that. So serendipitous discoveries are retrospective analysis also we can use. That is we call as nothing but the de novo methods. Okay. Repurposing drug could be key fighting antibiotic resistance. Definitely repurposing will be the great, the great kind of great revolution in the field of pharmaceutical drug discovery process. There are the few examples of repurposed drug. We all know the first drug which was repurposed is sildenafil, originally intended for hypertension, but repurposed treatment of erectile dysfunction later on. Thalidomide, which was originally intended for morning sickness, but repurposed for treatment of leprosy later on. Recent example, metformin, hydroxychloroquine, dexamethasones, various drugs are there, which have been developed for different purpose, but now it is being used for different indication. That is clinic to lab. That is the translating research. You all of you know reverse pharmacology. You have to always moving toward the from classical to forward pharmacology to reverse pharmacology. Many things are coming, indication coming from the lab, clinic. So clinic to lab, that would be the reverse pharmacology. You have to think about that. There are different drugs which have been, approved for new indications. So what strategy we can use for drug targeting? So just look at this as an example, okay, how we can repurpose the drug. For that, one has to understood the drug mechanism, how your drug is acting on the particular receptors. In case of bacteria, what are different target site of bacteria? So all of you understood target site of bacteria. It could be efflux mechanism, it could be the inactivation of proteins, it could be the inactivations of this cell wall. Various approaches are there. So how we can repurpose the drug? Recently, just I have taken one example because I work on the curcumin along with the piperine. So all of you know the curcumin is is a natural phytoconstituents. So we know the mechanism of drug like amoxicillin, ampicillin, which is mostly getting producing resistance because of drug efflux mechanism. If we take the amoxicillin, ampicillin, a drug, that antibody become resistant toward the species like Asorius and Pseudomonas species. But if you use this, curcumin along with the piperine, which is the PGP inhibitor, because this is the efflux pump that is called as PGP inhibitor. We have different influx and efflux proteins. This is the efflux proteins, PGP inhibitors. Because of that, drug is coming out. Once the drug is come inside the cell, because of this efflux protein, drug is coming out. It will throw out the drug outside the cell. So drug will become resistant toward that particular protein molecule. So if you use this piperine along with that antibody, definitely we can control that efflux mechanism and drug resistance. We can resolve that issue and one can utilize the same drug, same antibiotic for repurposing for the same. That is a kind of approach we can use. So I have been work on that. Definitely, I am also getting so many things on that. But only thing is that we should understood all this mechanism at molecular level. Then and only we can come up with the various solutions. One has to understood that. What is the conventional approach? What is the bioinformatics approach? Now look at the conventional approach. What we target in serendipity way, means without knowing the target, without knowing anything, we just do the serendipity study. We do the kind of search study. In case of conventional approach, then we get the proper molecules, active fraction, then we go with the peptide identification, that is protein identification with your sophisticated technology like NMR, crystallography, MSLS. Then we perform in vivo study, and then we'll get all these things. But if you use in silico approach, what is in silico approach? A simulation approach, computational modeling. You have to apply some proper algorithm in order to screen, in order to find out the proper lock and key. Means we have the key, that is ligand, and we have the proteins, that is we call as lock. We have to find out which key is going to fit it with respect to their conformation, coordination, and proper pose. Pose is very important. That is the theory behind this molecular docking. We understood lock and key theory, then we understood induced fit theory, then we understood conformational selection theory, that is population shift, where we consider the free energy, relative free energy and binding sites, electrostatic and van der Waals forces are being involved. So that is the kind of understanding in case of virtual screening, that is molecular docking. So where we use this can be utilized over here. No need to go directly over here. This will save your time, because if you go with this way, it will take 7 to 8 years. But if you go with this approach, definitely you could save your time. Same thing can be done, get it done within 1 year, maybe 6 months. That is the beauty of this bioinformatics approach. These are the software engineering experiments. If you just talk about the normal process, in vivo, perform in vitro study, then we go with in vivo study, then further clinical trials with human volunteers. But this is the new concept coming in virtuo, that is virtual screening, and in silico approach, which is need for software engineering knowledge is required. We require some algorithm, which we have the sound knowledge of, all algorithm, all computational modeling, all scoring function, all your search algorithm, what are different search algorithm we are supposed to use, what are the scoring function that will give the predictions on interaction of energy correspond to the each prediction being confirmation. That is the need of our, that need to understood. This again classical to modern approach. I already talked on that. Okay, one has to understood this homology modeling, means similarity study. We have the protein bank. Only we have to do the similar. We have to find out which particular protein is getting similar with your ligands. That is called as homology modeling, protein-protein interaction, machine learning, molecular dynamics. That is nowadays coming many you all of Gromax and Schrodinger's. All softwares are being used for this molecular dynamics, virtual screening, more docking simulations. All these softwares are available. I'm going to take you on that also. Don't worry about that. But these are the things we should understood. Bioinformatics, immunoinformatics, pharmacogenomics, which is the future of the pharmacy, we could say, where we are moving toward the personalized medicines, genomic medicines, precision medicine, we could say. Pharmacoinformatics also one of the area, fascinating area, where our student has to focus. And next, what are the drug repurposing flow? How it has to be? For that, we must understood the pharmacokinetics. So already the known pharmacokinetics, because we have taken the FD approved drug which have been appear in the RAE book as per para 1, para 2, para 3, para 4, we understood the regulatory affairs. So we have to take that drugs for which is being approved for clinical use, then which have been passed by safety trials, that is phase 1, but failing demonstrate efficacy in phase 2. Then drug replaced by better therapeutics and drug become generic due to patent expiry. These are the drugs which have been available for repurposing. We can use that. We can use existing compound also for getting novel target approach. It is based on the observation that many drugs bind to multiple targets. One has to understood that this secondary target could be the related to the different disease of physiological condition that we understood in hydroxychloroquine. Now people have found that it has been bind with that, that ACE2 inhibitors. Because of that, we are using that hydroxychloroquine for COVID-19 prophylaxis periods. Then known mechanism, if the mechanism is known, it is based on the observation that several biological process and signaling pathway are relevant for more than one disease. Hence, the same drug which inhibit the biological process that can exert effect on two different disease states. That is the idea how we can get. So for that, we can go with the data mining, omics. Again, omics come into picture. So we have the mechanistic approach, descriptive approach. We can use that approach for finding the mechanisms, because that will reduce the risk, cost, time, and polypharmacy. That is that are the benefits of this repurposing of drug. We can integrate into therapeutics. This is the one example. Recently have been appear into markets, not market, but it is in the clinical trials. Okay, the first drug which have been completely done, completely made, prepared, or we call discovered by artificial intelligence. The world first medication or medicines which have been created by British startup Excensia and Japanese pharmaceutical firm Sumitomo. The drug name is DCP-1181, which will be used to treat patients who have obsessive compulsive disorder, OCD. Typically, drug development takes about 5 years to get into trial, but the AI, that is artificial intelligence, drug took just 12 months. This is the best example of pharmacal modeling. So what is pharmacal modeling? Means the basic moiety which have the biological activity. So we have to find out the basic moiety which is having a biological activity that will give some therapeutic indication. That is called as nothing but the pharmacal modeling by using computational tools. So this would be the first drug, I could say, which have been developed by artificial intelligence. This is in the clinical trials. Now current medicines, one treatment fit for all, but future medicine would be the personalized medicines, which will be giving the proper effect on human being. Because we are considering their genetic characters, genetic makeup. All vaccines, biosimilar product, nothing but the vaccines or other interferons and monoclonal antibody, which is called as biological products, which have been developed for biosimilar. The pharmacogenomics. What is bioinformatics now? This word have been coined by the Pauline and Beck in 1970. It is nothing but the use of computational tools. Computational means nothing but the molecular biology, or we call as computational technology, tools to discover new information in complex data, from one-dimensional information of DNA to two-dimensional information of RNA and three-dimensional information of protein to four-dimensional information of evolving living system. All of you know the COVID-19, which is the the refined version of SARS-V1. We all of you understood how scientists got to know this information because of bioinformatics, because they understood fully genomic sequence of that COVID-19. Because of that, we could get all the information whether that particular microbes have been evolved from the earlier system or not. That kind of things we can get to know from these bioinformatics tools. This is kind of evolution in the field of pharmaceutical drug discovery process or drug development process. So this is the emerging branch, all of you know, the field of science in which biology, computer science, and IT, information technology, merge into single discipline. Biologists and pharmacists collect the molecular data, DNA, protein sequence. We have the huge database of DNA and protein sequence. Gene expression. We have the Human Genome Project. We all of you know. We can collect all data, retrieve all data. And computer scientist, what he does? It's a mathematician, statistician, develop the tools, software algorithms to store, analyze the data, and bioinformaticians just to study the biological questions by analyzing the molecular data. That is use of work get it done by bioinformaticians. Let us take the example of this. Okay, we have the database now. Database, we have the DNA RNA database, nucleotide database, protein database. Now what we have to do? We have to take the known and new proteins which is having similar to that proteins. So that proteins you have to run. You have to just put that protein into that database and we have to see the alignment similarity in genomic sequence, that is, which is the complementary sequence with that existing protease. That we can get it done in database. We have the database, PDB database, protein database, series database. The database are being useful to see the similarity and complementary evidence, complementary sequence with the existing database. Definitely your work would be reduced. And that information is gathered from bioinformatics and can be used to identify and to get to know the evolution and history of organism. You can get to know the history, evolution of that microorganism, how this microorganism have been evolved, like COVID-19 2, how it has been evolved from COVID-19 1, SARS-V1. That we can get to know. Only thing is that we have to understood genetic algorithm and stochastic search methods. That is very, very important. Stochastic search method is nowadays used for genetic genomic process. Maybe next speaker will talk on that, I don't know, but definitely this will be the area where we can talk. We have target site, we have ligand site. Ligand is your drug. We have to do the docking study. We have to just see the fitted model, best fitted model, with the help of their conformation, coordination, and energy level. What is Pharmacal model? Nothing but this word have been developed by Paul Ehrlich, you all of you know, father of chemotherapy, which he developed the first kind of chemotherapeutic drug, which is the sulfonamide derivative. That is the first fully synthetic drugs, which were developed by Paul Ehrlich, who is the father of chemotherapy. At that time, the understanding was that uncertain a chemical group or function in the molecule were responsible for biological effects. With the similar effect had similar function in common. The word pharmacophore was coined much later by Schuller in 1960 in book Chemodynamics and Drug Design, was defined as a molecular framework that carries, that is phorose, that essential feature responsible for drug pharmacone biological activity. Means which molecule get properly with your log, that is nothing but a pharmacal model. It's a biological moiety, a basic moiety, okay, which is having a biological activity. We call it as nothing but the, as per the IUPAC definition, pharmacophore is the example of electronic feature that is necessary to ensure the optimal supramolecular interaction with a specific biological target to trigger a block biological response. We all of you know, either it go act as a stimulation, act as inhibition. We all know receptors, drug acting. We all know agonist or antagonist. We understood fully all that. I'll take you on this now. Pharmacophore fingerprintings. Okay, already we discussed with that the approach that being used to for pharmacophore modeling. Either we know the receptor, either we know the ligands. There are, if you know both the things, like ligand is known one and your proteins are known, then we can go with the structure-based drug design. We all know structure-based design. If receptor is unknown, okay, but if you know the your ligand, then we can go with ligand-based drug delivery system. If you known and ligand is unknown, then we can go with the de novo methods. We all of you know de novo method means complex to simpler form of molecule we are making. For that, one has to understood the Lipinski rule. That is very, very important. One has to understood what are descriptors playing role. We all know we understood Lipinski rule. What are the criteria required for drug molecule to have a biological activity? The first is log P value. What is log P? Log P is nothing but the partition coefficient. So that should be always less than 5. Then only the drug is having a drug is called as drug-like characteristics. We can call it as a drug. Then another is your molecular weight. It should be always less than 500. In Daltons. That is very important. If your molecular weight is more than 500, then drug is not going to be act on your receptor, not going to produce any kind of prominent biological activity. The next would be your hydrophilicity. That is also playing vital roles, because hydrophobicity and hydrophilicity is also very, very important. We must understood that. Okay. Then next is your hydrophobicity, that is CH3, C2H, H4 and all molecules, which is complement to receptors. Homology modeling, then cationic anionic groups. Then we understood aromatic rings also very, very important. That is aromatic ring. Then hydrogen bond acceptor should not be hydrogen bond acceptor means O and N should be you all of you know less than 10. Then donor should be less than 5. They are the criteria as per Lipinski rule. One has to understood that, because we understood all this BCS classification, biopharmaceutical classification, log, where we consider all this solubility and lipophilicity. We always understood class 1 drug is most prominent drug. We all know which is having high solubility and high lipophilicity. So that is the thing one has to understood. How we can do the modeling? So I am taking you this. Already we discussed about this Lipinski rule. This is very important. Your drug must be having these all the feature. Then we can say drug is having that characteristic or drugability characteristics. I will take you on Zinc Farmer. There are a number of softwares are there. Gomax are there. Atodox are there. Golds are there. Schrodinger's are there, Zinc Farmer are there, Paymol is there for molecular visualization, and number of free softwares are there. Because in this 1 hour, it's not possible to cover each and everything. But definitely, I will take you on Zinc Farmer. Okay, how we can do the drug drug docking and molecular docking for drug discovery process. I hope you all are getting, okay? Directly zinc farmer. Yeah, I hope. Yeah. Yeah, Pawar sir, is it okay if I continue with the next 30 minutes of 20 minutes? Hello? Pawar sir, am I audible?
Yes, sir, you are
Okay.
audible.
Yeah. Shall I continue next 20 minutes? Okay. How much?
Yes, sir. No problem at all, sir.
Yeah, thank you. Thank you for your consent, sir. Yeah. This is Zinc Farmer. It's a free database, a web-based application, which is having multiple a number of tools are there, but it's very useful for me for my study, where I could come up with the various solutions. We can identify the pharmacophore features. For molecular docking and simulation required near about 5 to 6 hours to go with one by one. Maybe 1 or 2 days required to go with that. But now this 1 hour not possible to all. So that's why I'm taking the directly which have been covered all the aspects. So I'm taking on kinase inhibitors. You all of you know kinase inhibitors, which are often used for treatment of cancer and inflammation. Zinc Farmer is the automatic pharmacophore features. Detection was applied to the inhibitors of PDB3K5U. These are proteins, inhibitor proteins like PDB3K5U. Because we have to identify the proteins of interest. That is very important. So protein making is one of the crucial steps in molecular docking. Protein making. We have the ligands, multiple ligands. We have the known proteins. Then definitely we can target on that. If you don't know the protein, then you have to make a proteins, prepare the protein, identify the proteins. By using crystallography, we have NMR technology. Through that, we can find out the proteins. Recently, we have 2 proteins in COVID-19. We all of you know 6LU7 and 7BQ, which is the M3 inhibitors proteins. This has been identified by various scientists, which have been deposited on the PDB data bank, which is for COVID. But because of time constraint, I could not continue with that. I'm taking this kinase inhibitors, which are often used for this. Okay, and this is a resulting screen identify the zinc. These are the promising ligands, which is annotated as a potential tyrosine protein kinase inhibitor, because they found very good energy level. We mostly expect more than -8, because minus energy is important, because more the lesser the energy, free energy, higher would be the therapeutic activity affinity toward that particular proteins that we expect. That is called a scoring function. The scoring function, the various factors affect, maybe force field, mechanistics, in order to get a possible freedom of energy. Low energy we expect, because all of you know the initially what happened, ligand have higher energy to impact on your receptors, because of the van der Waals forces, because of electrostatic forces. But later on, whatever energy is going to be minimized. So that minimized energy we are taking into consideration as per the a conformational selection theory, which was proposed by some scientists in 1978 onwards. So look at this. I'll just click on this. Okay, now kinase inhibitors. Okay. So how we can, for that we should make ready your proteins of interest and ligand of interest. Then we can load that feature over here. Option is there, load feature. So because of time constraint, I could not load, but already which have been loaded that I'm taking. But feature is there. We can add a feature. So the strategy would be you have to select descriptors. What kind of molecule you are going to make, and what could be your target sites, whether it is hydrophobic target, hydrophilic target. So that you have to decide. That is called a descriptors. You have to just think about descriptors, because these descriptors play vital roles. That is the fundamental information required to make a molecular docking. So always you have to take into consideration hydrogen donor, hydrogen acceptors, aromatic ring, cation, anionic structure, aromatic rings, and all this kind of we can say hydrogen bonding acceptor and donor already we take, hydrophobicity and all. So you have to take into consideration. We can do this. We can play with this. The strategy is required for that. So what kind of molecule you are going to make? Load feature means you can load your ligands, and load receptor means you have to load your protein of interest that you are going to target. In case of your COVID-19, we identified 6LU7 and 6BQ, which is N3 receptor binding sites inhibitor proteins. Similarly, for kinase inhibitor is one of the target site for your drugs. Now load receptor already I have loaded over here. Now see, you could see here the 3D structure of proteins as that is called as ligand. Ligand is there, have been entrapped properly with these proteins. That is lock and key again. You have to find out the conformation and coordination, orientation, proper pose, then submit a query. So there are a number of ligand proteins are there. You have to find out the best fit model for your drug, for your ligands. Proteins are there. Okay, now you have to find out which is the best ligands molecule that is going to fit it with your proteins. Now see that they are searching 206433075 confirmation available in the Zinc Farmer. The database, huge data have been uploaded. Now you have to think, you have to utilize, retrieve the data for your purpose. Now it 1 or 2 minutes, within a 1 or 2 minutes, you will be getting a most promising molecule, which is having that, RMSD, that is root mean square deviations. Yeah, we call it freedom of energy, that you can take it into consideration. Now see, 7 hits are there. Still it is being going on. Now see, the 19 hits are there. Means we can find out the hits, which is the hitted molecule, that we can see lead optimization and hit molecule is there. No? We do in serendipity study, but here we could do it within a one minutes. You'll be getting n number of hitting. Like see, within a 51.83 second, we shall able to find out 228 hits. If you go with normal process with your lab, a wet lab study, it will take near about 1 year to 2 years. But if you within a span of very few minutes, a second, you could say, you'll get the all hits. That is the beauty of this computational modeling. You have to find out the proper algorithm is very important. Now see, we can take the best fitted model. What is RMST? That is nothing but the we call as root mean square deviation, which is having less energy that you have to take it consideration. Now see, less energy I could find over here 391. So I can take this molecule as a promising molecule for my particular kinase inhibitors. Now see, this would be the best ligand for my. Likewise, we can do it. You can make a structure, 3D structure. Only thing is that you require 3D structures. We have drug data bank, you have PDB data banks. We can take it from that. Now see, which is the structure. Now see, this is the promising molecules, which is having all the physical characteristic. Now see, X log P we consider less than 5. We got here 70. A polar means nothing but the polarity, hydrophobicity 3.57. Hydrophilicity is minus 17. Hydrogen bond donor would be less than 5. It is 1. Acceptor less than 10. We got 6, which is best molecule. We can say net charge 0. This is the topological molecular weight less than 500 as per the standards. We got here 276. Rotable bond is 5. So they are the property that we got it from this. So we have this is the molecule of interest. We can take it into further clinical trials. So that is which is having identity of 99.90% with the similar your ligands. This is the kind of things we can do it. I hope you understood this. This is the kind of beauty of Zinc Farmer. We have the PDB database for that. If you want to get to know about proteins, we have the protein database, PDB database. Okay, PDB database we have. Okay, RC. You can note down the website over here also. rcsb.org. This is the if you want to get a protein. Now recently we have facing the problem of COVID-19. Now see, recently this molecule have been updated over here. Even you can also update over here. We can also submit your proteins of interest proteins to this database. Now see, recently the June molecule of month, recently somebody has submitted this SARS-CoV-2 spike proteins. Definitely you can find out your existing antibiotic, existing drug for getting a new solution for that coronavirus. This is the kind of beauty we can do it. Now there's no possibility to go into wet lab study or to go with your lab. Definitely this pandemic situation, we can utilize your time for doing such kind of work. That is called as a dry lab study. Wet lab is not possible at this moment. However, we can use these all kind of tools. Now see, this is the molecule of interest. We can see if you want to find out any kind of new molecule like 6LU7. This is the 6LU7. This is the protein inhibitor proteins. Okay, 6LU7. Now see, this is the protein of which act against COVID-19. We can get to know this particular protein. Now see, this is the proteins. Now 3D structure is there. We can just download these proteins and we can run your existing drug. That we can do it. These are crystals because there's crystal structures. So what is the limitation of this docking study? Only thing is that you have to remove the heteroatoms. That is very important because we are doing the crystallization process and all the NMR technologies. For that, cofactor is going to remove. You have to remove the water molecule. Because resolution is very important. Your molecule should be highly resolved, more than 2 angstrom. We expect more than 2 angstrom. That resolution is expected. Then and only we can get a good affinity towards your molecule. That is limitations that we using this kind of strategy. Always remove heteroatoms. Okay, that is not needed because that will create a problem for docking study. This is the crystal structure of COVID-19 main protease in complex with inhibitor N3 proteins. These are proteins. We can get to know all these things. Okay, we can get 3D structure. You can copy it. You can download it. Option is there, and you can run on Zinc database. Gromax, Schrodinger's are there. Zinc Farmer are there. We have Atodox is there, which is commonly used. All are free softwares. Schrodinger is not free software. Gromax is commercial software, but we can use all that. Okay, they are providing 1 month free access also if you just approach to them. I hope you are getting all these things. Then PayMol also there, one website. PayMol is used for molecular visualization. If you want to see proper visualization of structure, then we can use paymol.org. This is the kind of, I'll show you this. I hope you all are getting it. Okay, definitely I'll answer all the questions. If you have any questions, any doubts, I'll be happy for that. But let me finish all this topic. Yeah, this is the computational modeling. I'll show you within 2 minutes. Yeah, how it has to be done. It's a 4 minutes video. Please look at this.
In molecular dynamics, atoms are typically represented as single point masses in their van der Waals potential. What were these 612 potentials typically used to represent this thing? This means that they come out mostly as hard particle charges can be calculated by some form of quantum method to represent the distribution of electronic charge on those molecules. For a polar molecule like water, these partial charges can actually be quite large, with the best part of a full formal negative charge on the oxygen atom. In molecular dynamics, bonds are typically represented as simple harmonic oscillators, as are the angle constraints. To start the simulation, a range of velocities is chosen that matches the Boltzmann distribution for that temperature, and then those velocities are assigned randomly to each atom. Similar methods are then used to put energy into the bonds, angles, and dihedrals. The simulation is then run according to Newtonian physics, with the molecules transferring both energy and momentum to one another via electrostatics and van der Waals interactions. So in these simulations, water is essentially a hard sphere with 3 point charges in it. Surprisingly, such a simple method for modeling these systems gives surprisingly good results on numerous physical properties that can be calculated. Maybe the best of these is the structure. So if we plot the average probability of finding an oxygen in a unit volume with distance out from a specific water molecule, we get what we would call a radial distribution function. Now, it turns out these structural functions can actually be measured directly via experimental methods such as neutron scattering. As you can see, these very simple methods do an okay job of modeling the structure of water.
Yeah, this is the kind of this we can hypothetically we can get to know. Okay, hypothetically we have to what the data we are getting hypothetically and predictable value that we can compare with this, because validation is very important. I would suggest use 2 or 3 softwares to confirm your all the data which you have been getting from your molecular dockings. That is very important. Need to validation is very important. We can compare with the standard moiety also. We can take some standard drugs for get to know about the information, all that, whether we have done it properly or not. That is because it could be reversible, irreversible docking process that need to be standardized. Validation is very, very important. That is we call as a scoring and post processing. So molecular docking can be done by target proteins, ligands. If both are known, then we can go with the structure-based drug design. If you don't know the ligand, then we can go with ligand-based drug delivery system. Then we can system preparation. For that, we need to understand descriptors, which descriptor playing vital role for your biological activity. Then we can score docking, because docking score is very important. That is scoring function can be done by force field, the possible freedom of energy. Mostly we expect low energy. That is we call a scoring function. Scoring, it could be the search algorithm. We have to find out the affinity and avidity toward your proteins. Then post processing. They are the steps involved in molecular dockings. Okay, there are a number of software we use. Okay. The Gold is commercial, Atodog is freely available, Zinc Farmer is freely available, PharmaGist also there, PharmaGist. Okay, I'll just take you on. Next would be this is the process already I have told you. Okay, sorry, docking can be done by docking process and reverse docking. Okay, in docking process, protein of interest we are putting into your database, then database will give the possible ligand binding. Reverse docking, what we do, we take the active component or existing drug, we put into your database, and we'll be getting protein of target database. Similarity, complementary proteins will be getting, and we can get to know possible binding proteins. That is the kind of approach we can use for docking process. It's a kind of virtual screening. We could say virtual screening. It's a virtual screening. Like clinical trials we have been doing virtually. Similar kind of virtual screening can be done in molecular docking by using computational tools. They are the free software tools for molecular modeling and docking and visualization of structures. We can take the snapshot if you want. Definitely, I'll be sharing this data to you also. This would be really helpful to you. Okay, so if you can visit this VLab, Amruta lab. Okay, because of time constraint, I could not continue with that. But VLab is where, Amruta lab, they have created the virtual lab where we will be getting n number of moiety, n number of things we can get to. Even we can explore to your students, M-Pharm students. Genomic sequence annotation we can get to know from this ASP.AHABS. Okay. Then Opel Babel also there is a free software we use to search, convert, analyze, and store data from molecular modeling to other associated areas. They are all free. Okay, all are freely available. Only thing we have to explore all these data. And structure of drug molecule, we can get it from drugbank.com. This is the one website where we can get to know the particular molecules if you want to know the information of particular molecule. Okay, so there are different software which can be used. Okay. They are docking study how we can get the score, because the proper scoring and posing is very important. Confirmation, coordination of your molecules with your proteins, that is very, very important. Now if you just look at this example, HI1 protease, there are different ligands are being there, different pose are there, which is the best fitted model that you have to consider. Okay, the scoring ligand which is the best, like pose 1, pose 2, pose 3. Okay, which is the best, having the best score with respect to the confirmation coordination, that we can take into consideration for further clinical trials. So they are the very important slide, I could say. Already I have discussed with this which molecules are violate the rules on Lipinski. That is already we discussed. H bond donor, acceptors, molecular weight less than 500, log P also less than 5. Okay, these are the violation of rules, which is poorly soluble. These are the rejected molecules because they have less water solubility. These are the many rings, like you all of know, inorganic and heteroatoms need to be removed. Then only we will get the proper confirmation coordination, and we can do the simulation properly. Heteroatom is not needed in case of docking study. Heteroatom is like hydrogen, nitrogen. We have, all of you know, your aliphatic rings and all, we can say water molecules. That is not required because that is used in that cofactor is using for crystallization purpose, but that need to be removed from your database. They are reactive groups, toxic groups. They are all multiple chiral center we expect. What are biological and biosimilars? Many people are get confused about that. Biological preparation means nothing but the biological means which is obtained from the biological source, made from living organism, to the highly complex manufacturing process, like your monoclonal antibody, vaccines, which is having it includes a wide variety of products such as genes, cell therapy, therapeutic proteins, monoclonal antibody, and vaccines. Biological are used to prevent, treat, or cure variety of diseases, including cancer, chronic kidney disease, diabetic, cystic fibrosis, and autoimmune disorders. So biosimilar are highly similar. They are not similar. They are not identical with the biological, but they are very similar. Like generic are highly, it's a copy of innovator product, but biosimilar are not a complete copy. It is a highly similar. Maybe they are structurally some different. The reference products in terms of safety, purity, potency, but may have the minor difference in clinically inactive components. The biological products, when it get expired by patent, appear in the purple book, while generic drug appear in the orange book, that we understood in regulatory guidelines. So if you want to work on biological, you always refer the purple book. You'll be get to know which drug have been expired by patents. So now I think 17 blockbuster biological drug have been expired by patents. Definitely the Indian pharmaceutical industry getting a lot of avenues or opportunity to come up with this kind of tools. Already had been started. Chanta Biotech, or Ipca, Dr. Reddy, your Sun Pharma already started the working on biosimilar products. So definitely the future is very bright for pharmacists, where we can work on these biosimilar products in order to make this product available at affordable price. Like generic product, we make it available at affordable price. Similarly here, biosimilar products are very costly. All of you know, biological are very costly. Like anti-cancer, you all of you know anti-cancer drugs, very costly. But one can make a biosimilar products that will be useful to all society. It will be affordable to all. That is very, very important. There are different techniques where we can use to understood the 3D structure of proteins. Okay, this is my work, turmeric. I've been working on turmeric. There are different software already we do. Okay, understood glides and Vina, Itodoc, Ledoc, Gold. All these softwares are being used to understood the binding affinity and interactive modes. It can be do with the reverse docking and search algorithm function. Already we discussed on that. So molecular docking could be by using searching function and scoring function. It could be target flexibility or ligand samplings. So there are different criteria being used. Okay. What is reverse vaccinology? This is now being used for biosimilar making developments. So normal, if you go with the normal vaccines, which is being used the whole organisms. But nowadays the scientists are being using the only gene of interest, which is having the antigenicity property. This is a gene of interest. Now see the one molecule, one microorganism, they're having different gene of interest. that need to be recognized. That is the promising, we can say, one of the challenging tasks for development of vaccine, a biosimilar product. One has to identify the epitopic sites we normally call as epitopes. Epitopes means nothing but the receptor site which are present on your called as antibody, sorry, antigens. Paratopes are present on your antibody. Okay, epitopes are present on antigens. They are the antigens. Antigens are nothing but the any kind of microorganism, any kind of proteins which is elicit that immunogenicity property. This is the different kind of proteins one has to identify. The protein can be identified by various sophisticated technology. Once to identify proteins, then you can incorporate penless. You all of you know why vaccine become penless, because we are taking only the fraction of very specific parts which is having the property, biological activity that you are taking, instead of taking whole organisms. But normally, if you go to traditional methods, they use all whole microorganism. It could be killed one virulent species. If you take whole microorganism, then that vaccine become a painful. But now we are moving to the penless drug delivery. So thanks to this all computational model and genomic sequence, we could understood, and we are coming toward this all reverse vaccinology process. This painless to painful to painless drug delivery systems. We understood here. Antigens are there. We have different epitopes. Now COVID-19 virus have different epitopes. There are about 22 epitopes have been identified. That's why it's very difficult to make a vaccine on that. Why the vaccine development is time consuming? Because the viruses are very highly resistant, highly we call it as mutation. There are a lot of insertion, deletion, mutations are going on. So it's very difficult to recognize that particular proteins of interest with this kind of technology. Okay, already we discussed with this. Okay, I'm not talking on that. There are different databases where we can use. If you want to get to know about nucleotide sequence, we can refer this NCBI website. Enzymes is also used to get the human genome informations. PubMed is widely used for literature references. Then protein sequence, then we can get to know from NCBI. Then SwissProt, which is used for protein sequence. Protein domain can be get to know from this website ebi.ac.uk. Then genetic disease can be get we can get more information on genetic disease by using this website, this link. If you want to get to know more about enzymes, because enzymes also play vital role for drug targets and all, so for that we can refer these websites. Protein just now we visited that rccb.org for protein structures, where we understood we can get updated information of proteins. There are about 1.5 lakh proteins have been deposited. How much? 1.5 lakh proteins. Nevertheless, we can use it for further exploration. Metabolic pathway we can understood through this genome.ad.in. Okay. Different databases are there. Some are sequence database, some are functional, some are literature, some are structural, specialized. So structural database are being useful for for getting for repurposing of drug and all. It could be secondary, primary, and specialized. So mostly specialized database are useful, and which is highly curated, because that is very curated. Means we can get proper very specific information. Primary database we have gene database, European database, all PDB, NDB. We have near about 3,000 biological database, vary in size, quality, coverage. It is very comprehensive and accurate, up to date, good interference, batch search, download. We can do multiple things. Only thing we have to explore. Exploration is important. This is NCBI website, how it looks. Then this is the entrance PubMed's website, where we can get to know various things. DDBJ data banks from Japan, where we collect DNA sequence located in NIG. Then this PIR, protein information source, we can get to know. This enzymes which are used for. Then these are the UCSC genome browsers, where we can get to know about genomic sequence of each and every molecules. National Center for Biotechnology Information, NCBI is very good tools where we get multiple informations. Then this is the website for that. Okay, how there are different features we could see at your extreme left corner. Then the database will we get to know different proteins of interest and all these things. So in probes we can get to know. Okay, these are the how we can get the informations in different sequence. PubMed is all of you know. You might have used the PubMed for very good search engine where we get all database. This is the PubMed's how it looks. Okay, homology modeling we can do it over here. Homology means similarity, complementary proteins we can find out. Okay, this is the molecular dockings paper. This is the enzyme research sequence database. Okay, these all are websites where we can use for your study purpose, because all are useful. These are all European database. DDBJ also, which is where we can get many information on genetic informations. DDBJ also is UniProt also being used. It's a manually annotated and non-reductant protein sequence database, which combine information extract from scientific literature and biocurator evaluated computational analysis. It provides all known relevant information about particular proteins. Annotation is very important. Annotation is regularly reviewed to keep up and current scientific findings. Okay. These are the UniProt website, how it looks. That these are the different proteins, PIR proteins informations. Okay. These all circular database are there. We understood PDB. Okay, where we get the 3D structure of all the proteins, because 3D structure protein is very important. We can obtain the 3D structure by X-ray crystallography and MR spectroscopy. There are two promising technology we use to get to know about 3D structures. That 3D structure we can submit to the PDB bank also around the world. It is freely available tools we can use. Okay, this is transferred to Research Collaborative for Structural Bioinformatics, RCCB. Their database, how many databases are there? You can see protein structure 95,636, the molecule which is obtained from X-ray diffraction. So they are the different analytics. Yeah, this is the protein data bank. Just now we revisited this PDB data banks. We can get 3D structures, and we can get to know the resolution also, how much resolution of them. Now we can see resolution is very important. 1.6 angstrom is quite good. Okay, actually we expect more than 2. Then only we can do the molecule docking properly. Okay, now combat drug resistance. Always, if you know no action today, no cure tomorrow. That is, we have to think about that, how we can go with this and cope up with the challenges. Finally, I concludes the virtual, finally, I conclude this. You all of cloud-based services need to be developed for analysis, real-time monitoring, antibiotic sensitivity. Computational modeling would be the one key of indicator for repurposing of drug, and virtual innovation lab to radically transform how future medicines and health services are developed and delivered. That is, need of our in order to cope up with the challenges with this. The other references and bibliography that I have referred for collecting all informations. And thank you to the team for conducting this very nice seminar and all these things. You have to use antibiotic very rationally, judiciously. No action today, no cure tomorrow. So we have to mold the future to be intellectually sharp, mentally alert, and spiritually elevated, and physically fit. Then and only it will be possible. Thanks to once again all for patiently hearing. Now I'll happy to take a question. Go to organization.
Thank you, sir. Thank you very much. Now I would like to invite the moderator of the session, Mr. Vipul Jain, sir, to conclude the session.
Once again, good morning to all, honorable speaker and the participants. In order to conclude the session, definitely, sir, whatever information you have been shared with our participants and with the students will definitely help them for the upliftment in the area of drug discovery. However, sir, you have been nicely covered the various topics such as a need of a journey from painful to painless medication, future of medication, making of future medicine with the concept of molecular biology, then discovery of the potential antibiotics, and also with the descriptive drug discovery and the development process. How bioinformatics database is useful for the drug discovery and the development process. All these topics you have been covered very nicely, sir. So now this session is open for the questionnaire. So if any participant is having any question, then they should please mention that question in a live chat box. So we will present or highlight that particular question on the screen. Once again, I am repeating. Now the session is open for the questionnaire. So if any participant is having any kind of question, so they should mention that particular question in the live chat box so that we can present or highlight that particular question on the screen.
Yeah, I could see some questions. What is reverse doctor? Shall I take the question from this, sir? Yeah, you are taking.
Sir, I will present that question one by one.
Sure. Thank you.
So this was another question from Mr. Akash Jain.
What you think about if you will added the other any active substance or other agents with antibiotics, then can we prevent? Yeah, definitely. This could be the one prediction hypothesis. Okay, hypothesis. That is my hypothesis. because all of you know the mechanism. First, one has to understood the mechanistic approach, how the drug is going to be resistance, how drug is getting, providing that resistance. If you know the mechanism, because of the PGP inhibitors, because of the efflux mechanism, your drug is, producing resistance. We can block that, efflux mechanism, and definitely drug will remain in your system and it will give the biological effect. That is the solution for that. It's a hypothesis. One has to understood, one has to do all the clinical studies further. But this is a hypothesis we can predict. We can get to form this computational modeling. Yeah. Any more questions? I hope you got the answer.
Okay, sir. Thank you. So one more question is there. This was in the question from Mr. Abhishek. And the question is here on the screen, sir.
Yeah, my question is how bioinformatics help in personalized medicines and what will be the regulatory impact on the personalized medicines using bioinformatics? So yeah, I think it's a very good question he has been asked me. Definitely bioinformatics tool have a potential promising, you can say, applications. Only thing is that, okay, so one has to identify, one has to study it properly with the clinical indications. We have the tools, it's a hypothesis. Again, you have to prove it with clinical. A clinical study is required for all of this. Okay, because we are moving the personalized medicines, we have to understood the whole genetics makeup of the individual person. Then only we can target the particular person, this particular moiety, whether it is useful to that particular patients or not. Because many people are responding to penicillin still. Why they are responding to penicillin? One has to identify. Maybe their genetic makeup is different. One has to understood. Some people resistant to the ampicillin. Some are not resistant. So when has to understood all this data should be gathered from clinical parameters, maybe in the clinics. So we have to make this database properly. So definitely we can come up with this kind of personalized medicines. But with the future, and regulatory is all of will be getting all the these things. Yeah. Next question.
Okay. So this
Yeah, I think I have answered these questions. Yeah. Next questions. I'm happy to, I'll be available on the WhatsApp. My personal number also I'll share. Maybe email ID I'll share. Definitely, you can also personally, you can chat with me. Not an issue. Because of time constraint, maybe we could not take all the questions. But definitely, I'm here to resolve all the queries. Yeah, next question. Is
sir,
there
this
any
was
free...
the question.
Yeah, is there any free software to perform docking study of group of chemical towards specific? Yeah, sir, software is there. Like zinc farmer, Atodox. Zinc farmer we can use for the chemical towards specific receptors, because we can load the receptors over there and we can study. So options are there. We can load a receptor, load a ligands, and you can get to know. You can also create your own database. Okay, but database is there. You can use that database for that. Zinc farmer is there. Pharmagist also. I would suggest go with Pharmagist and zinc farmer. Freely available. Yeah. Yes, sir. Next
So
question.
this was on the next question from Dr. Sachin Satak.
Yeah, thank you, Sachin sir, for asking very nice questions. What is the success rate of into successful drug? Sir, if you just see the drug from the CADD, the first drug was norfloxacin. All of you know. Now later on, near about more than 100 molecules have been developed by using this remdesivir also. Remdesivir also obtained by bioinformatics only. Dexamethasone, how they are got to know within 1 or 2 months that drug is useful for that. So it has a lot of potentials. Only thing we have to explore all this database. Yeah. Next question, sir.
Okay. Thank you, sir. The next question. This was in the next question from Honorable Poonam Vasista.
Yeah, I think this is the very basic question she has been asked, but that is a need of hour. If you want to really, if you have will, we can make a way, find out the way. That is willpower is very important. Your attitude and all is very important. The skill set is very important. What skill set you should have? You must understood first mechanism, all molecular biology. What is the mechanism of that particular kind of therapy? How the drug is acting? What is agonist? What is antagonist? Okay, one has to understood descriptors. What are descriptors are there? What are Lipinski rule? Basic fundamental is very important. If your fundamental is clear, definitely we can do. I am not, I'm not from pharmaceutics, from chemistry. I'm a pharmacist. I'm working on all the aspects. If you just look at this, see, only you should passionate about the work. That is very important. The skill set is basic, fundamental. The molecular biology, biochemistry, microbiology, biotechnology is very important. And physiology also. Yeah, thank you. And chemistry, that is very important.
Okay, sir. Thank you, sir. This was the next question from Honorable Sunita Garge.
Yes, IC50, madam. For that, we have to perform this study in vivo study only. But competition modeling, we can't get this kind of value IC50 and all. Then QSR practical, because this is a practical approach, wet lab study. So here we'll be getting hypothetical prediction. See, you need to understand this all computational modeling are the hypothetical prediction. So that will predict, that will give the closeness to your your kind of hypothesis. That is the beauty of this computational tools. Okay, again, you have to validate with the clinical trial. Clinical trial is required for that, because unless and until you do the clinical trial, you won't come to know IC50 and all these things, because that is very important. But we can get to know hypothesis. This is the drug dose. This is the solubility. Based on solubility, ADMAT study, we can predict how much dose required to get that therapeutic activity. That we use that Gastro Plus and various softwares are there. Okay, Gromax also will give the idea on that. Yeah. Next questions.
Here is the next question from Honorable Dr. Sandeep Pathare, sir.
Yeah, thank you, Dr. Sandeep Padare, sir. You are listening me. I think since last 1 or 2 months, you are listening my all lectures. Thank you very much for asking the questions to me. So, what are the basic metabolic differences one can consider to find a drug target against pathogenic organisms? Very good questions. He has been asked me, because metabolic difference is very, very important, because we are talking about proteomics, genomics, another area where we can understood metabolics, because that is very important. One has to understood the metabolic pathway, how drug is acting on your receptor sites. What is the metabolic pathway? Different sequences are there. If you understood the mechanistic way, mechanistic approach play vital role over here. One has to understood how drug is, in case of COVID-19, how your drug is acting on how your pathogen is, replicating by different steps. Now why scientists understood that ACE2 inhibitors? Because they understood that your virus is going to be attached on your mucosal surface, where that ACE2 inhibitors are there, that play vital roles. Then next would be after incorporation into your cell wall. Then next another, it start replication through certain genes, certain proteins and enzymes. We can target that enzymes. Likewise, we can go. One has to understood the mechanistic approach. So mechanistic approach play vital role over here. I think this is the very good questions he has been asked me. That is, we can target. So each and every level we can target. You have to take into that all the sequence in loop, and we can target which promising molecule. You have to just do the virtual screening. Maybe we'll get good serendipity. Yeah, thank you. Next.
So a few more questions are from Dr. Pathare, sir. The second one is now.
Is there any way to predict oral bioavailability for natural product component using computational models? So hypothetically, we can predict, but that is not 100% sure that we'll having but definitely with the help of their all physicochemical parameter like log-P. From the log-P we can consider, we can get to know, log-P value is more than 1, 2, 3, 4. Definitely you can predict drug is having high lipophilic characteristic. Definitely lipophilic drug only going to be absorbed, permeate to your cell membrane. Then log-P value, then molecular weights, all these prediction, that will give predictions. We can compare with standard drugs. If you have standard drugs, you can compare with that. Definitely your idea will be, your hypothesis will be clear. That is very, very important. So computational modeling definitely will give the hypothesis on that. We have the tools. Gromax could be the one of the best tool for that. We'll be giving even Gastro Plus. Okay, yeah. I think, I've answered all these questions.
Yeah. One more question is from Honorable Yogita Pathare, ma'am. Here is the question on screen, sir.
Can we predict the drug resistance in HIV using bioinformatics? Yes, ma'am. We can predict. If you know the mechanism, like again, whether it is efflux mechanism, whether it is inactivation of enzyme, that one has to understood. Now you know the enzyme structure. You take that enzyme structure in silico library, and if you are ligands, you can just run the screening. You will be getting the best fitted model. We can target that. Definitely, we can do it. We can. It is possible. Nothing is impossible in this world. Word impossible, say I am possible. Okay, but you should be perfect in that. So perfection is very important. Practice make man perfect, and you should be always do the perfect practice in order to make it perfect. Yeah.
One more question, sir. There. Last question. This was from Tejaswini Girase. The question is there on the screen.
Thank you for very good questions he has been asked. Like you told, MDR as a threat to mankind. Is there any alternative way we can go in the future? Definitely for that, what about we can repurposing is the one solution, repurposing of drug, because we cannot go with the new novel drug delivery process, novel drug discovery process. It's time consuming and slow pace is required. If you go with the repurposing of drug by using computational modeling, bioinformatics tools, definitely your problem will be solved. again you have to do some campaign in the society. You have to make awareness about this. You have to use rationally, judiciously these all antibiotics. Then only it's all. Yeah. Thank you once again.
Okay, sir.
Yeah.
Thank you, sir. Thank you very much. The questionnaire session has been get closed now.
Yeah.
But sir, few things which I want to share with you, the comments which we haven't received on the live chat box. That comments I want to share with you, sir. The comments are such
Please.
as a very excellent presentation, a very informative session, a useful session, and lastly, sir, there is a huge demand of your PowerPoint presentation from our participants. They are demanding for your PowerPoint presentation.
Yes, definitely, I will share that, but there are some copyrights are there. I will just remove that, and
Yeah,
definitely
okay.
I'll. Sure, definitely.
Okay,
Definitely.
fine, fine.
But it should be used for personal use, not to be commercial purpose. Yeah, that is the only thing.
Yeah, definitely. So thank you, sir. And now I would like to invite once again Honorable Dr. Sunila ma'am for the introduction of our second guest, Honorable Dr. Yogeshwar Bachau sir, and to continue with the second session of webinar. Thank you.
Thank you, sir. Thank you, sir. Now we start the second session of this webinar by Dr. Yogeshwar Bachau. He is a founder and director of ADEX Pharmaceutical Consultancy Services, Mumbai. Here, I want to introduce this dynamic personality before the startup of this session. I'm very proud to say that Dr. Yogeshwar Bachau is an alumni of our college. After finishing PhD in pharmaceutics from the Institute of Chemical Technology, Mumbai, he was working in Europe, BACH region for around 14 years with different startup and mid-sized biotech pharmaceutical companies. During this tenure, he has successfully managed pharmaceutical development of several projects from POC to phase 1, phase 2, phase 3 clinical trials. He has strong network of experts and CROs in Europe, USA, and Australia. In addition, he has 16 international publications, 8 patents, and 26 conference proceedings to his credit. Recently, he has edited a book for Ville VCH, Germany on innovative dosage form, design, and development at early stage. He is a founder of the pharmaceutical R&D consultancy firm ADEX Pharma, which was established in 2006. Currently, he is serving as Associate Director at AI Curies Anti-Infective Cures in Germany. Now, I request Dr. Yogeshwar Bachcha sir, please take the charge of session.
Okay.
Please, sir.
Yes, sure. Good morning, all. I just want to make sure if I'm audible, you can all hear me very well. I'll start to share my screen.
Okay, sir.
Yeah.
Just
sorry. Can you see the screen?
yes, sir.
Okay. So once again, good morning. Many thanks, Professor Sunila Patil, for very kind introduction. Many thanks, Professor Sunil Pawar, for giving me opportunity to be here. It has been really a privilege if I have this possibility to talk little bit in front of my teachers, my fellow colleagues, and the juniors from my alma mater. Nothing greater than that. So in today's topic, in line with the theme, I would like to talk about the topic drug development process and CMC challenges. Of course, Dr. Patil already tried to introduce me, but I'll just like to give a few more details. So as said, I'm very proud alumni of College of Pharmacy, Shada. After that, I did my master's in medicinal chemistry with Professor Kamanchi, PhD in pharmaceutics, Professor Vandana Patrauli from University, nowadays ICT Mumbai. And then in 2007, I moved to Switzerland. I started working with the startup biotech company, Pantic Biosolutions, and there my job was to develop the medical device for transdermal delivered biologics, and there we managed to move from the proof of concept to first successful pregnancy in Czech Republic. So it was quite nice experience how you see really concept moving from, let's say, academic atmosphere to the commercial world, or at least to the clinical phase. And there we really work with peptides, proteins, antibodies even. And then I moved to Swiss pharma company Debio Farm in Lausanne. I worked there for 4 years as a formulation manager. And the company, the company's focus was really to develop new oncology drugs. It was quite nice experience when you work with different kinase inhibitors, notch inhibitors, try to see how they are moving really from the docking studies to the lab studies, and then you go on into the clinic. And then I moved to a German biotech company, Icuris, which is in fact spin-off company from Bayer. So, and there I started working as a head of pharmaceutical development. Again, this company is mainly focused on anti-infectives, antibacterials, antivirals, and it is again a lot of fun working with new molecules, really see how they are really coming from the lab experiments in the let's say in antibacterial field. Naturally, ideally you really start with the even petri plates and try to see how you are moving the species like mice or rats, and show on how it is going into the clinics. I've been still working with this company as a full-time consultant because for private reasons in 2016 I was moving to India partly, so I'm still shuffling between India and Germany. So I manage my consultancy firm at the same time. I'm still associated with Icuris to manage their pharmaceutical development activities. So having said that, with this background, I tried to manage little bit to compile all the information, how the process of drug discovery and drug molecule development looks like, what is the typical CMC challenges. In terms of bioinformatics, I think Professor Bolshetiva really gave excellent presentation. So maybe in the interest of time, I will not repeat the same things, but I will try to correlate how the drug discovery scientist or as a formulation scientist like me, I'm looking for the bioinformatics for the key information. So regarding the contents of my presentation, I'll give a brief overview of drug discovery process, how we try to understand the disease, how drugs are acting, how the process of new drug development looks like when you go for target identification validation, followed by lead optimization. Then you start with preclinical, clinical phase, go for FDA approval. I will also touch few examples how bioinformatics is helping in drug discovery process. And what are the CMC challenges? CMC means chemistry, manufacturing, control. And this is something quite important in the process of drug development. What typical impact it has on drug substance, drug product, and then I would like to conclude that presentation. So let's start with the new drug discovery and development process. So why the drug discovery is really essential in today's era, if the question is coming. So we do have some unmet medical needs. The best example I can give you, nowadays we do have the COVID-19 epidemic, and everybody's asking when the new drug is coming, and that's where I see unmet medical need. You have a lot of cancerous diseases. Cancer is a broad group, but there are a lot of subtype of diseases, and let's say for many indications we still do not have successful drugs. Same is case with Alzheimer's or AIDS. We are truly missing a drug. Nowadays there are a lot of incidences of infections like bacterial infections, which are like multi-drug resistance. There is no, let's say, super antibiotics working against all the bugs, and the treatment is really missing there. So these are the unmet medical needs, and that's where the new drugs are needed. Sometimes the cost of therapy is even very high, and it may not be affordable, especially for the developing countries, and that's where there is a need for the new drugs. And again, the drug development companies need to sustain in this atmosphere. As I said, I've been working with drug development companies. They're putting a lot of money on the table. So at the end of the day, they are also expecting some revenues. And this is how they would like to channelize the resources and the financial, let's say, the income in order to continue the drug discovery process. On this slide, you can also see the chart or the graph, how many drugs are being approved from 1993 until 2019. And this also includes the biological entities as well as a new molecular entities. And on your right, you see the CDA approval trends, especially in 2019, how it is being increasing if you try to compare from 2014. So you do have some priority applications, some orphan indications, some accelerated indications. So when you try to see the priority application, imagine if any company is developing any drug for any infections where there is no alternative treatment, they can always ask regulatory authorities, look, this is the drug, currently no treatment available. Can you just try to put it this priority application? Because this really helps to speed up the FDA review process, and maybe drug can be brought onto the market very soon. So it is a little bit overview of the drug discovery process, how it starts, what type of disciplines are involved, what it takes when you try to take a decision to initiate the new drug discovery process. So there's a lot of scientific and technical exchange, of course. So because you come up with some hypothesis, then you try to make sure what kind of target you are trying to identify, whether you have any in-house assay models, what kind of animal models you are going to use in terms of chemistry, how it looks like, whether you have the possibility to get a very powerful pattern where it can be sustained, what is the competition landmark. Imagine you are a small company and you have Roche or Novartis in front of you, and this is also one aspect you always need to consider. And in terms of development, what are typical challenges? Often in the drug pipeline, you see a lot of molecules which are not quite easy to be developed, and formulation could be a showstopper. This is something you need to really identify in advance. Toxicology, again, we learned that using the genomics, using bioinformatics, you can really predict what kind of toxicological effects you can really imagine from these molecules. This is also to be taken into account. In terms of strategy, very important. Imagine I'm working with iCuris and their focus is really anti-infectives. So they will never go for cardiology for sure, because you need that kind of team of experts who can really understand the disease mechanism, how you like to plan your preclinical and clinical trials. So this is very unique for every company. They will always like to focus with their potential interests. In terms of operational, you really have to see what kind of staff you have, because imagine you are working with any biotech company, you just have like 30 to 40 people or maybe 50 to 60 people. In that case, you can't really imagine to have phase 3 trials. You can really start with the preclinic or early research, maximum at the end of phase 1 or phase 2. You will be looking forward out licensing your molecule to the big pharmas, and then they go for phase 3 trials. Timelines are always important because you are always looking to your competitors. Imagine you are having competitors who is really running faster to launch their drug. So all the efforts, what you have put, all the money you have put on the table, it's really going to waste. So this is also something you have to keep in mind. So what it takes for the successful drug development. So in the graph below on this slide, you can see the average time needed to develop a drug. So when you see the drug discovery, you really start with the 10,000s of compounds. When you go to preclinical, you may have like 100, 250 compounds. With the clinic, ideally you start with one compound, maybe you have some compounds as backup candidates. You go for review and then drug is approved. So it's overall process of 10 to 12 years, having investment of almost 800 million to $1 billion. That's what it takes. And for this, then it's very important you do have the state of art technology on your side. The project management is also complex because often the clinical trials are running around the globe, and you need to coordinate that because any single mistake can really cost a lot, especially when you are really into the late phase clinical trials. Being a CMC project manager, I can tell you often you are running your drug product manufacturing sitting somewhere in India, maybe the campaign is running in Australia or US, and that's where the project management is really coming into the picture, that virtually you need to monitor a lot of things, and which is important. Again, the resources, it's very important because resources may be in the form of the team, that you need a dedicated team of scientists for that. A lot of financial aspects are also involved. If I can give you any biological molecule you are trying to develop, maybe protein, maybe vaccine, a simple GMP production for phase 1 trials costing almost 8 to 10 million US dollars. So a lot of money you are putting onto the table. So you also need to take care that companies financially, very well settled. They are able to manage the funds. As said, the scientific minds are very important. Very high attrition rate. This is something very important to keep in mind, because as you clearly see on this graph, you start with 10,000 compounds, and by the time you are reaching phase 1 trials, you may have only one compound. So you can already see the attrition rate there. And this is something to be always kept in mind, what type of risk you are taking there, because the economical risk is there. Imagine one of your lead candidate is failing, maybe at phase 1 or phase 2, then a lot of efforts are getting waste. So this is something to be avoided. And as said before, it's a time consuming process. So the level of patience should be very, very high. And here you see the phases of creation of the new drug. So typically you start with the drug discovery, where you try to see really how the concept is moving into the molecule. Imagine if I have to develop any anti-infective drugs, there I try to see which typical receptor I need to target. Maybe that receptor is sitting onto the cell wall. Maybe that receptor is really involved into any, DNA transcription or RNA synthesis, or maybe the release of viral particles from the cells if I'm developing any antiviral molecule. So this type of concepts you try to imagine from the pharmacological point of view, and that's where you come with some potential structures or potential mechanism. And once you have the molecule, you try to develop that molecule into the registered product. But before that, you really need to go from the process of preclinical and clinical investigation, which certainly we will discuss in the later part of the presentation. And once the product is approved, then you go for commercialization, because then you go for to the FDA with your NDA dossier. FDA reviews its safety and efficacy, and based on that, they try to approve that product. Then it goes to the, let's say, to the market, to the sales. And in the graph below, you can clearly see then how you're moving from concept to target selection, followed by validation of the target, lead finding, lead optimization, clinical development, then phase 1, phase 2, phase 3 clinical phases, followed by regulatory approval. And that's where you go to registration. And again, once again, it takes around 10 to 12 years for a normal development. I think it's more or less same information I said before, but I think it's very important to keep in mind, at what phases of this development the role of the CMC scientist, medicinal chemistry, or pharmacologist is very, very important. So for each phase, you can really see what is important. So at preclinical phase, you really try to check whether your drug is effective in that animal model, whether it's toxic. Based on that, you know already what dose you want to select for a phase 1, and that's where you go to identify the safety of your compound in the clinical disease. In phase 2, you try to check how efficacious your drug. And once you have that information, you try to dare, I'll say dare, because for phase 3, you really need a dare, some sort of daring, whether big or small company, you can really try to go for phase 3, or maybe approach big pharmas that phase 3 trials can be initiated. And there the job is really to check the safety and efficacy of your compound in large number of patients. And once phase 3 is successful, you go for FDA review, and after approval of your drug, you run phase 4 trials. And in the table below, you see the number of patients you need, the length of duration. So phase 1 trial, which are also often called as first in man trials, they're limitedly small trials, so done on, let's say, 20 to 80 patient, takes 6 months to 1 year. Phase 2 is relatively bigger. You go maybe around 200 to 300 patients, takes 1 to 2 years. Phase 3 is relatively large. Also depends on the indication. For some indications, maybe you can go with 1,000 patients. Some indications are too complex, time consuming. Maybe you need higher number of patients, but it also depends on the regulatory guidance. And then you go for filing. So maybe when you start with the drug discovery project, when you are trying to imagine what kind of target you are going to use for your drug development, maybe the human genome sequencing is also very important. This is something completed in 2001. So far we have around 20 to 25,000 genes already, let's say, identified. They're made up of 3 billion individual base units, base pairs, which are the DNA units. And each particular gene, it codes specific proteins, or it could be enzyme, which carry out several functions to the body. They're also part of the disease pathologies. And that's why this knowledge of genetic sequence is very important, because finally they are going to code the proteins which are really involved on this disease pathophysiologies. So even though we do have this genetic information available, but it's very complex to know about their interaction or their specific participation into the disease pathophysiology. And that's where the bioinformatics is coming into the picture. We can really use this database, what kind of proteins are being expressed. They are really linked to which, pathophysiological state, whether overexpression or lesser expression of these proteins is leading to any particular disease. And this information is very, very important when you try to start your drug discovery process. So let's try to discuss a little bit how the drugs are acting, or when you try to identify a target. This is something very important. You need to find out how the particular drug is going to act. So in this example, I tried to summarize, especially for the cardiovascular indication. You really need to see if you are targeting any drug against vasoconstriction, vasodilation, what kind of receptors are involved in that particular process. And once you have the knowledge of those receptor structures, then with the computational methods, you try to find out the 3D structure of that particular receptor. You go for docking studies and see if your potential structure is going to bind. And that's where I'm again coming back to you. It's very important. You must have clarity in your mind how your drug is going to act. On your right, you have example from the tuberculosis model. So you must try to at least summarize or try to imagine how this mycobacter species is going to infect the human cell walls, what kind of genetic material is going to be replicated into the human cells, and that's how you really try to see how each and every individual drug is going to act. Maybe it is acting onto the cell wall, the peptidoglycan receptor, you can imagine. Maybe the drug is going to act on the RNA polymerase, and once you have those typical structures, then it's maybe then this is a starting point. This is how you try to come up with your potential lead candidates and try to see if drug has any potential to bind to that particular receptor. So I said before, with this knowledge, then you try to come up with potential lead candidates. You try to come up with some chemical structures. So with the help of the modern technologies by MRI, using X-ray crystallography, the let's say computed drug design, the medicinal chemists, they try to come up with, let's say, chemical structure because they already have the 3D structure of the target. They try to perform some docking studies to make sure how your drug is binding to the receptor. At the same time, you really want to make sure that the drug is very specific. It is not binding to the normal receptors. Imagine you are developing anti-cancer drug. The job is really to kill the cancerous cell, but not the normal cell. Then you have to really make sure that drug is really going to bind to the receptor, which is expressed into the cancer cells, not to the normal cell. And that's where these docking structures are docking studies are very important to ensure the drug is specifically binding to that particular receptor. And on the top of that, of course, once you have the docking, the recent advances like combinatorial chemistry really helps to come up with the synthesis, the library of compounds, so that you do have the possibility to screen the several hundreds of thousands compounds at the same time. Because alone docking is not going to give a compound for further development. You really need to check your in silico results with actual results, and that's where you need to perform the screening. So I think I already talked a little bit before that how disease identification, understanding of that particular disease is very important, but maybe we can discuss a little bit more. If I am trying to develop any drug molecule in cardiological indications, then we should try to understand more in detail how the disease looks like, what kind of pathophysiology is involved in that particular disease, what kind of receptors are being involved, which we are going to really target. And that's why the careful understanding of the disease is very important. Same is the cancerous cells. In that case, you should try to understand a little bit more whether that particular receptor is being expressed onto the cell surface, whether it is going to be intracellular, whether your molecule is going to penetrate. Imagine maybe you are trying to develop the drug for a brain cancer, and in that case, you should be at least able to understand whether the molecule you are developing is able to cross the blood-brain barrier. If it is not the case, then all the efforts you are putting may not be successful, and that's where the proper understanding of the disease is very important. If I have to summarize, let's say in case of antibacterial drug development, so it's very important there whether I want to develop a drug which is going to act onto the cell surface, whether it is going to penetrate the cells, or whether it is going to act into, let's say, bacterial cell replication. So sort of understanding helps a medicinal chemist, pharmacologist to create a mechanism action of drug. That's where you try to really give some input that what kind of drug molecules we are really targeting here. So in this slide, let's try to understand a little bit more about target discovery. So what kind of targets we are really targeting? So here you see some summary of the different human targets we do have. It could be GPCR, G protein coupled receptors, ionotropic receptors, especially the cancer receptors like kinase receptors or the Notch receptors. And then this really, there's lot of database already available. Maybe we already tried to learn a little bit more into the previous talk, and this information is always very helpful for the drug development scientists in order to check which receptors we have to target. Often you come with some novel mechanism action, so these are like first in class. So you have like two types of molecules, it may be first in class or best in class. So when it is first in class, you really come up with a novel mechanism of action. So maybe you are not able to find that particular receptor database. So you have to come with your own hypothesis, your own mechanism of action. But having this understanding of the target is very, very important. So when you say human targets, in that case, especially we are talking about the targets which are normally present in the human cells. But when you are dealing with infectious diseases like antiviral, antibacterial drugs, so these are not human target. These are the receptors which are really present onto the bacterial cells or maybe viral particles. And again, having this information is very, very important. At least you know which targets we are going to target there. And what are the strategies we are using to find the new drug targets? So of course, in that case, you can already try to check how the approved drugs are being, uh, how the approved drugs they are were acting onto the receptors, whether that interaction is strong enough, or whether we have any possibility to increase the efficiency. Maybe you can take the pharmacophore of that existing drug, try to do some structural modification by SAR studies, QSAR studies, and see if there's an increase in potency. This could be one approach. You can also try to see the mechanism of action of the existing drug. For example, aspirin is working on COX, but there's also a role to play, or there's also some way you can find out how this COX inhibition is helping in other way, and that's the reason you can already see the action of aspirin in some other diseases. It's quite widely used even for the cardiovascular indication. These days, even I saw some clinical trials where aspirin was working against migraine. So this type of information is always very crucial when you try to come up with new targets. So once the target is selected, of course, the job of medicinal chemist is really to verify how this target is involved into particular disease pathophysiology, how it is going to, how the drug is going to bind to that target. And in that case, the validation of target is very, very important because you have to verify really that particular target is really involved in that pathophysiological indications. And that's where scientists also try to ignore or avoid some paths because then chances of failure are quite high. The dead ends he may meet are very high. And that's the reason the validation of target is very, very important. And for this purpose, we need large number of complex expense trials because this is not only in vitro or in silico studies. You have to also find out in vivo how it is going to act and whether you are able to get the desired efficacy, what has been really planned into the drug discovery process. And these are the different aspects of target validation. So you should know what kind of animal model you are going to use for disease. So the questions are there already. I can see some of the webinars people were asking what kind of animal model we should really use for COVID. The clear answer is still missing in that case. So this is very, very important that whatever target you are being highlighted, you are being, you're going to highlight, and during the validation, you have to make sure that this target is already present in the preclinical model because that gives you sufficient confidence to go into the clinic. And even you go for dossier submission, at least you have to propose a mechanism of action to the regulatory authorities because that also helps them to understand that drug is really going to work through that mechanism. There are no expected adverse events, and that's where they try to minimize the risk into the clinical volunteers. So I think identification of biomarkers is very, very important. And this is typically important. I mean, I can share from my experience in Debbie Farm where we were developing the oncology drugs. The role, the finding typical biomarkers is very, very, very important because that really tells you how your drug is efficacious into the clinic. The overexpression or inhibition of typical biomarkers goes hand in hand with the potency of your compound. That's why it's very important. Often you do not have the possibility to get the in vivo models. In that case, the cell-based disease model is very, very important because there are a lot of ethical consideration you might have. It's time consuming. So having cell-based model is always very, very important. And often the disease association genetics is very, very important because at the end of the day, all this information is always going to help that how your drug is acting to the target, what kind of genetic alteration it is having. That really gives significant input if you want to go to the next level of compounds. So with this knowledge which we have gained through the target identification validations, then the researchers, they have sufficient information for the search of potential drug candidate chemical structures, and they try to really focus that the drug is going to act onto the target and it is really going to have impact on the disease pathophysiology. And as said before, after years of research and plenty of hurdles and obstacles, if successful at all, then the lead candidate becomes the drug molecule. And how we go for the lead candidate identification, so there are several ways. So you can always look back to the nature is greater source, always the greater source of the drug molecules. As we know, drug like penicillin, they are coming from the natures. You do have some molecules coming from the moles. Phytochemistry is also one source of the molecules. The de novo approach is something very simple, where you try to search the drugs from the scratch. In that case, again, computer modeling is very helpful because it helps to predict some active molecules. High throughput screening, very important tool, because it allows to screen the hundreds and thousands of compounds against the target. Genetic engineering is very important, because of all the proteins, antibodies you see onto the market, even into the pipeline, they are basically coming from the genetic engineering of the living systems. And I think one thing we should really discuss, all the computer aided drug design we have discussed so far, it really works well for the small molecules, but what about the large molecules? Because often this question must be coming, and your Professor Polshet, you will agree with me. All the computational studies you might do during the docking studies, especially I'm talking in terms of physicochemical properties, maybe pKa, log P, it works quite well for small molecules. But what about large molecules? No? So this is also one aspect very important to consider. It has to be addressed, and some work is definitely needed, that when we are trying to develop the complex biological molecules, how the in silico tool is really helping the formulation scientists to rule out any failures in advance. So let's discuss a little bit of typical high throughput screening process. So in that case, as you see on your right, on your left, sorry. So I think you have three aspects to consider: time, quality, and cost. So this high throughput screening really allows you to do a lot of tests very fast, because you are trying to screen the several thousands of compounds into using the plate reader, maybe ELISA plate you can consider for the example. And that's where you really try to see that how you can do a number of screens in the shorter time frame, and that really helps to decrease the time you need for the typical project. From the quality aspects, you have to always pay attention if you have any false positives, false negatives, how many hits or leads you may have, because overall aim, you do not get all the leads. Again, first you start with the hits, and once you start with hit optimization process, you try to go to the lead. And then the cost, that because if you go for high throughput screening, you try to do a lot of expense to in parallel, you try to do in shorter period of time. So definitely the significant reduction of the cost. So in that case, when you go for high throughput screening, you must have suitable assay in place. That assay must be validated, and that assay must be able to show you that you are able to get the robust pharmacological response. And during this process, you go for primary screening, so where you do have large number of compounds at that particular concentration, and this process gives you the hit compounds, and heat is confirmed, then you go for second round of testing. You try to screen those hit compounds at fixed concentration, maybe in duplicate or triplicate, and this really allows you to optimize your potency in order to check what IC90, what IC50 is expected for that compound. When you try to say the target product profile TPP, you already know against the competitor product onto the market what IC50 or IC90 you are aiming for. And if it is not achieved, again you will go back to the structural modification. You try to run the series of screening, and finally you will stop once you have the desired IC50 or IC90. And once you have the lead compound identified, and then the efforts are really focused on the lead optimization, because in that case you have to really make sure that your compound is really active, it's not causing any adverse effects, and that's where the safety aspects are coming into the picture. So there the medicinal chemists, they try to alter the chemical structure of the compound through the modification of the, let's say, functional group onto the molecule, that leads to the change in physicochemical properties. Maybe it's more hydrophilic, maybe it's more lipophilic, maybe the pK change, the binding constant is changed, and that's where it results into the let's say higher potency in that case. I mean, this is also one way how you can reduce the adverse effect of your candidate by minimizing their interaction other chemical pathways. So in simple words, you are trying to make your drug candidate as specific as possible. In that case, you try to come up with hundreds or thousands compounds as initial leads, and then they are synthesized, they are tested. That's where you know whether your drug is cytotoxic, whether it's active or not. The pharmacologists and chemists, they really try to work together in this process, because pharmacologist is the person who is going to check these compounds in the biological systems or assess them, and they try to give the input to the chemist, and chemist really tries to see with what structural variations if, they see some typical higher or lower antibacterial antiviral activity or different pharmacological response, and that's how they do the structural changes. And after all this assessment, you do have the drug candidate, or the, let's say, the candidate nomination text test when you want to initiate your preclinical studies. But at this early stage, there's very close interaction between the even CMC people like us, because we have to also check if the molecule they have proposed is also, feasible to be synthesized, whether there are any formulation challenges. Imagine you do have a compound which is working excellent, no toxicity profile, but at the end of the day, as a chemist, if I'm not able to synthesize, as a formulation person, I'm not able to formulate it, then what we are going to do with that drug? So this is also one aspect we have to consider. So briefly, how this computer-aided drug design or CAD is really helping into drug discovery. I just tried to summarize. I said before, I mean, we had excellent talk from Professor Polshettiwa, so it doesn't make sense to repeat everything again, but that's how the CAD is coming into the pictures. You start with the target identification, where the analysis of gene sequences, the protein structures, metabolic path is very, very important. When you go for target validation, then the similarity studies are very, very important to check how your drug is going to bind to the receptors. Then docking studies also very helpful to identify the lead candidate. And finally, for the optimization of your lead, the QSAR studies are very, very helpful. And once your lead candidate is optimized, once it is nominated for development, you go through the typical process of drug development. So this is a brief overview of how you really go from the preclinical phases up to the clinical phase at the end, because these things we'll be discussing several times. That's the focus of this presentation, but worth of discussing. So you start with the lab test. If it works, you go for research protocols, you go for typical animal studies where you want to check your safety as well as the efficacy of compounds. When that also includes GLP tox studies, and based on the GLP tox studies, you already know what dose of the drug you are going to use in phase 1 trials. Phase 1 trials, you try to check how drug is safely administered into the humans. There are no adverse events. In phase 2, you try to check, which are typically proof of concept studies. You really try to check your drug's efficacious or no. If the results of phase 2 are not very promising, nobody will take really risk to go phase 3. Better they will come back again to their research. They do some structural modification in order to make their drug very efficacious. And then you go for phase 3, which is in large number of patients, where you try to check the safety and efficacy of compounds. Then you have very detailed biostatistical analysis of clinical data, and based on that, FDA goes approval for the drug. So let's try to discuss clinical trials. So these are often done into the lab animals. So typically, when we do the tox studies, these are done in rodents and non-rodents, because you certainly need the data on 2 species before going into the humans. And their object is really to check how drug is very well tolerated into the animal species, at what dose you see some toxic effects, because that helps you to predict the NOEL, what you say, no observed adverse effects dose. That's called NOEL. And based on that NOEL, you try to predict the safe human doses. Along with that, you have to also check the pharmacokinetics of your drug, how very well it is absorbed, what is the level of distribution, how it is being eliminated. I can give example, some drugs that are extensively eliminated by kidneys, but then this is also linked to some, let's say, kidney related toxicities, and this is then big concern for the immunologically impaired patients who have some already difficulties with the kidney functionalities. And these are all the aspects we try to really discuss well in advance, because you don't want to develop a drug where you may see some adverse event into the clinic. And this is something to be addressed quite in advance. With respect to the pharmacokinetics, you have to also check whether drug is very highly soluble, whether it is permeating well, often the drug permeates well, but maybe there's a PGP efflux. So this is something you have to check whether it is going to have some drug-drug interaction at this stage. And this is something you try to address. Again, from preclinical studies, you also try to check what is the mechanism action of the drug. I mean, whatever you have really, you had hypothesis before, whatever you have predicted, whether you also see in vivo. And then you start doing your clinical trials. So typically you go from phase 1 to phase 4. We discussed before, the phase 1 you need less number of patients. Again, important to highlight here, the success rate of a phase 1 is almost 70%, because you just try to check there how safely drug is tolerated. And in phase 1 trials, you never start with your human dose. You try to do some dose escalation studies. Maybe it's a single dose escalation studies, maybe multiple dose escalation studies, and that really helps you to decide what dose you are going to use in phase 2 studies. And phase 2 studies are typically focused on the efficacy. There you try to check against either with the comparator onto the market. You may do some placebo control trials, but overall aim is really to check the efficacy of your drug. And if you really find that the drug is really efficacious, you are able to get the desired response, you go for phase 3 trials, which are really into the large number of patients. And maybe you have to do all over the world to cover different patient populations. Again, success rate of phase 2 is 33%, for phase 3 is 25 to 30%. Imagine for phase 3, the cost of development is really high. So one has to really think twice whether they have to go really for phase 3, because the risk we are taking is really high in that case. But it is successful, then you go for approval. After approval, you have to do some additional clinical studies, which are typically phase 4 studies. And there, of course, you have thousands of participants, because you have the large number of patient data pool there. And that really helps us to get a lot of important information, what really happens when the drug is being taken for the long term. So what kind of effects we are getting, what type of adverse effects we have, whether there's any difference in the different sub patient group of around the world. And this data is going to be very useful. So once you have successful phase 3 studies, you go with the IND application to FDA, and what they really expect at that point, because the IND dossiers is really expected to contain all this information, starting from chemical structures, the route of synthesis, stability studies for both drug substance and drug product. What is your formulation composition? What kind of stability data you have? What kind of primary packaging is available or being used? The non-clinical and clinical data package, the proposed mechanism of action, and the detailed clinical trial plan they would like to see. And they will also like to make sure that whatever volunteers you have used there, they are not exposed to some risk, because maybe in animals you try to see the toxic effects, but overall objective is really to check that there is no risk for the human volunteers which are coming to the phase 1 trials. So these clinical trials are often reviewed by institutional review boards to make sure there is no risk for the patient. They try to get the informed consent from the patient so that the enrolling is possible and phase 1 studies are possible. Of course, the both FDA as well as sponsor company, they can stop any trial at any point of time. And typically, this is linked to also any adverse effect. Imagine you have any IV drug which is being administered. Suddenly, you have some precipitation into the light, or there are some pain at the site of injection. That is also considered as a adverse event. And based on all this, I mean, I'm just trying to give you some example. Based on such events, then investigators reporting to the company is going to report to the FDA that trial has to be stopped on the immediate basis. So let's try to discuss a little bit more how these phases of clinical trial are being planned, what is important during these phases. So for phase 1 trials, normally it is done on the small group of healthy volunteers, but depending on the indication, often you do have the patients. For some oncology indications, even we have seen that the disease patients were enrolled. Even I have seen some HPV trials where the disease patients were enrolled for phase 1 trials. So there are some exceptions, but majority of the cases you do have healthy volunteers for phase 1 trials. So in this trial, the first objective is really to check the pharmacokinetics of the drug in vivo, because all the data you have so far, it's maybe in silico or maybe from the animal models. And then job the typical objective of this particular phase is really to make sure that how the preclinical data is translated into the clinic with respect to the pharmacokinetics. So you may have some different metabolism of the drug into the humans. Some metabolite may be toxic, you do never know. Your drug may have some drug-drug interaction with some drugs which are being administered concurrently. So this is something to be verified in phase 1. Phase 1 trial also let you decide what kind of dose is very safely tolerated, and based on that you take a decision what kind of dose you are going to use for phase 2 studies. And then after successful outcome of phase 1 trial, you plan your phase 2 trials. In that case, again, small group of patients, you try to use like 200 to 500. But in that case, you already know what dose you are using. And with that dose, you try to check what is the predicted mode of action, whether it's really fitting with the clinical outcome. And most important, you try to check how your drug is acting against the comparator. Comparator could be another drug onto the market, or maybe you try to check it with placebo to in order to make sure, in order to conclude that your drug is efficacious. It is providing a pharmacological response which is being mentioned into your dossier. And then based on the successful outcome of the phase 2 trial, you try to plan end of phase 2 meeting with FDA, and there FDA allows you to perform the phase 3 trials. So phase 3 trials, they are the most expensive and lengthy trials, so the risk you are taking is really high. So in this case, the investigators, they try to evaluate the compound in almost 1,500 to 5,000 patients. Often these are done around the globe, US, EU, maybe Latin America, Asia specific, in order to check what kind of response we are able to trigger into the patients. What is the risk benefit ratio? Because this is very important, because imagine you are, I mean, there's no drug onto the market without any adverse effect, forget it. So you're certainly going to have some adverse effect, and that is risk you are taking. And when you say benefit is what is benefit you are bringing to the drug. Let's say you are developing a drug against COVID, so that's benefit. So maybe your drug is slightly toxic, it is inducing some nausea, vomiting, these are typical adverse effects. But maybe imagine you do have some pain at the site of action. So these are, let's say, adverse events, these are being considered the risk. But against that, if the benefit is really high, the patient cure is very high, and that's where FDA tries to investigate the impact of this risk benefit ratio. And that's where the phase 3 trials should be carefully planned, because one has to really ensure that at the end of the day, this risk benefit related question should be answered to the regulatory authorities. So I said before, the again, objective of this phase is also to check how safe and effective your drug. And this also provides the basis for labeling instructions, because your label, if you have any drug, check the label leaflet. It always tells you the what dose it should be, how frequently it has to be taken, what are the counter medication we should be try to avoid. And for that, then this data is very useful for the labeling instructions. And then, as said before, these trials are often done around the globe with the hundreds of sites. So collection of data from the different sites is very, very important. The CTM supply, the clinical trial medication supply, is a really huge task. And for the small companies, sometimes not so easy. And that's the reason then very big pharmas are able to do these phase 3 trials. And in parallel to this trial, because of course we don't want to spend a lot of time after phase 3 trials for the filing, so often the efforts are already initiated in parallel where you are running in phase 3 trials. Of course, the pharmacologists, clinicians are involved there. The people like me, the CMC people, at that time they are really working into the scale up of API process, the drug production process, to make sure that all this information is available for NDA dossier. And then, as we discussed, that safety of the drug is very, very important. The safety package is part of your NDA dossier. So you have to really make sure that from in vitro skin. So these days, a lot of in silico tools are already available. They try to predict the mutagenic and cytotoxic, let's say, the profile of the drug candidate. So and this is something should be available. Then you try to generate the non-GLP, GLP toxicity data into 2 species, and that really helps to predict your doses for the clinics. And when you run your clinical studies, depending on the acute or chronic administration, sometimes even you have to generate the GLP tox data from 12 months to 24 months, so that this data is available at the time of submission. And this is a typical FDA approval process. You try to go with your dossiers. There's a review meeting followed By that, FDA takes around 60 days of time to review the dossier. They do appoint a committee of experts to check what potential benefit that particular drug molecule is bringing to the patient. They try to evaluate the risk benefit ratio. Based on that, this voting of the committee members. FDA is not entitled to consideration that committee voting, but often it's the case. But based on that, then FDA takes a decision to approve the drug. Sometimes they may need some additional information, or maybe your dossier is rejected if you're unlucky. But once drug is approved, then they really try to check the facility inspection that what your drug substance or drug product we are producing, this is in line with the compliance with respect to the quality. Then the drug labeling process is investigated. If there are any comments from the FDA, it should be taken into account. And then you go for filing. And once the drug is approved, it is launched. You need to commit to the FDA that we'll be doing some post-marketing surveillance phase 4 studies. And from time to time, you need to submit this data to FDA. And this is a common technical document, what we also say CTD, regulatory terms, which is to be submitted to FDA. It contains different modules. So module 3 is really important from the CMC point of view, which contains all the quality related information to drug substance and drug product. Module 4 and 5 contains the non-clinical and clinical data package. And briefly about phase 4, that as I said before, this is a post-marketing study. So in that case, already the large number of patients are taking this drug, and it's very easy to get that information from the clinicians. Periodically, you have to submit this information to the regulatory authorities. FDA may ask for additional phase 4 studies, and this allows you to really conclude what is the impact of long-term administration of your compound onto the safety. You can also check there what is the effect of different subgroup of patients. Maybe during the entire clinical trial, you may not know what is the impact on pediatric population, especially long-term use I'm talking. So this way, phase 4 studies really allow you to check, how drug is acting in different races, different locations, different group of patients. So this was all about preclinical clinical studies. Let's try to discuss how the bioinformatics is really trying to help the drug discovery process. So by definition, bioinformatics or bioinformaticians, they really deal with computational management, the analysis of biological information, which is genes, proteins, cells, ecosystem, medical information, and how we can use in drug discovery process. Because as I said before, drug discovery process is using the high throughput screening. So in that case, this molecular data from the bioinformatics is very, very useful, especially between symptom and carriers and the normal controls. And this bioinformatics can really facilitate the drug discovery process, because it allows us to identify the drug target quite rapidly. Then it also helps to perform the screening quite rapidly, as we discussed before in computer the drug design, how the 3D structure of the target is really helping us to check what kind of molecules are really going to bind, whether they are specific, how through the structural changes we can really improve this binding, and we do have the chances to increase the potency of that compound. It further, it also helps that if there is any incidence of side effects, because this mainly comes from the non-specific binding. So this is also something we can avoid using bioinformatics. If there's incidence of drug resistance, because of in case of antibacterials, already there are some efflux systems involved into bacterial cell walls, and that's how they're really pumping out the drug from the bacterial cells. And once you know that receptor, then it really helps to block that particular receptor. This is, let's say, the resistance breaking mechanism, if you can say. And this is something possible with the bioinformatics. So as sir said before, this high throughput data such as genomics, epigenetic, omics, we say in general, is really helping a lot. I'm sorry, I'm running out of my battery, but I hope it's managed. So this omics data is really helping a lot to come up with the screening of the drugs. In that case, we do have the possibility to contribute for the mechanism-based drug discovery and drug repurposing. This is little bit, the slide is a bit crowded, a lot of information there, but in nutshell, what I'm trying to say that this omics related information is certainly going to key. So let's try to take one example, this protein RNA structures. Once we know, it really helps to really check really what kind of binding and docking properties are involved. whether there are any charge residues available onto the receptors, and how it really helping to have the electrostatic interaction between the drug and the target. What kind of enzyme substrates are available there, and this substrates then give some guide for the medicinal chemist to come up with some typical structures so that it is possible to envisage or to predict what kind of ligand protein interactions we are imagining. And this is again about omics, that how this omics data is helpful in that case, because earlier what happened, you had all this data maybe as post launch, but nowadays a lot of focus is also being paid that whatever data we already have from the already approved drug after post launch, whether this can be already made available at the early stage, because based on that database it's very helpful for the medicinal chemist to check what kind of targets or what kind of at least lead candidate we should already avoid, because which might lead to the, let's say, wrong path or maybe dead end, where we can see maybe very good efficacy but the incidence of adverse events is very, very high. This is something can be easily avoided. So on this slide, I try to compile few examples of the successful disease control, which was as a result of the bioinformatics. So congenital myasthenic syndrome is one of the conditions where there are different subtypes, almost 12 subtypes. In that case, at least it was possible to have the sequencing so that they could really pinpoint the exact subtype of the disease, and then they really came with the precision medicine, or maybe you can even say personalized medicine. So, and as we have already been discussing so far, for the rational drug design, the almost 99% validation of the homology model of the Zika protein was done with this approach, and that's really allowed to come with the inhibitors which are in the preclinical trials. And even this technology is really helping with the design of drug carriers, which are the substance for the some receptors, because it really helps you to decide then what kind of carrier we should really try to use for the drugs which are really binding to the particular receptors. So I think regarding database, sir already said a lot, so I don't want to spend time on this slide. So let's try to figure out some CMC aspect we should consider, which are really critical during the process of drug development. So I think this Lipinski's rule of 5 is very, very important. When you have the potential drug candidate, then you really have to check the medicinal chemist or formulation scientist, whether it's, of course, it's not always possible, because I can give you example. You have cyclosporin like compound onto the market. It has a molecular weight in the range of 1,300, but Lipinski says that molecular weight of the drug should be less than 500. So it's not always possible to really match, but often it is being referred by the people, by the scientists, and we try to check that, because it at least give a good indication that whatever molecule you are taking for development, it has some drug-like properties, or it is let's say druggability. We also try to say druggability. So the log P should be less than 5. You do have, it must have a hydrogen bond donors less than 5. It must have hydrogen bond receptors less than 10. Molecular weight should be less than 500. So why it is called Lipinski rule of 5, though there are only 4 parameters, because it's in multiples of 5. So this is also one
Hello. Sorry for interruption. We are trying to connect soon.
Hello, can you hear me?
Yeah, yeah.
Hello.
Yes, sir.
Sorry. So I managed to log into my mobile. So can you just do a favor, share the slides which I have already shared with you before, so you can put on your screen. I think I mailed to Pawar sir and the general mail. So it'll be very helpful if you can open the slides and put on the screen. Sorry for this technical error.
Just a minute, sir.
Yeah, no problem.
Sir, just a minute.
Yeah, no problem. Take your time. Do you have some issues, or maybe give me 2 minutes?
All the participants are informed that the break of 2 minutes will be there. The session will be resumed after 2 minutes. Please cooperate.
Hello. Hello.
Yes, sir. Yes, sir.
You can
Your
hear
voice
me.
is audible.
Okay, good. I'll just open the slides. I can start. So I'm extremely sorry for the inconvenience. So, it's technical error. We can't really help out. So I really hope for your understanding.
Yes,
I'll
yes.
just
No
start,
problem.
start from the slides where we stopped a few minutes back. Give me 1 second. I'm just sharing my screen. Okay, so I think I was at Lipinski's rule. I'll just shortly share my screen. You just need to confirm if you are able to see my screen. Can you see the screen?
Yes, sir.
Just okay, perfect. Let's start. Okay, so I said before, I mean, we always try to verify, the drug molecule we are about to start with the preclinical investigation. It should be meeting the, let's say, the specific set of the rules proposed by Lipinski. So, and even there are exceptions. I said, if you try to imagine all the structures of the proteins, peptides, or antibodies, they will be never fitting into that. But nevertheless, at least the log P, because higher the log P, maybe if you have a very lipophilic compound, it will be very, very difficult to get that compound into the formulation. So at least some of the information is always important. So we can't just rule out. Then let's try to discuss a little bit of drug substance. What are the same aspects which really try to consider when we go for, let's say, lead candidate to the preclinical phase, or maybe from preclinical to the early clinical phase. So how many numbers of steps we have in the synthetic process? We had one compound where there was 22 steps of synthesis, and even you have problem with one particular step, it's really messed to sort it out, the synthesis. And if your supply of drug substance, drug product is delayed for any preclinical or clinical trial, then that has a big impact on all your timelines. So this is something is very important. Then what feasibility we do have transferring the process from milligram to kilogram scale. Often at very early stage, we may have the chances to purify the compound using chromatography, but this is something we cannot imagine the large scale. You can't really put the big chromatographic setup to get the kilogram of compounds. So this is to be verified. Then what is the possibility to get the raw materials, building blocks needed for that particular API? I can give a very good example. I mean, we all know for a lot of raw materials, building blocks, we are still dependent on China. But imagine a current scenario where there was COVID, now we have like war-like situation. So in this type of scenarios, we must have some backup plan. We must try to identify the backup supply so that the supply of the drug product is not delayed. Then what is the regulatory status of the key starting materials? Imagine any of your starting materials are carcinogenic. If it has any mutagenic potential and there is any purging of the impurities from the starting materials to the final product. So this is also something to be carefully, ruled out. At least we should have a proper justification for these impurities. Then what sort of stability data we do have for intermediate raw materials? So because often these materials are being shipped transatlantic or maybe trans-Pacific or maybe going from Asia to Europe. And during this shipment, there must be some stability issues. Maybe some impurities are generated. So we must have robust stability data available for this material. So the purging of impurities I already discussed before. We must be able to identify what sort of carcinogenic or mutagenic impurities we do have in the API. Then polymorphic form of the API is very, very important, because at the end of the day, we want to have the very stable and robust polymorph, which should not change during the storage or during the shelf life. If there is any change in the polymorphic form, certainly it is going to impact your drug product performance. It is going to impact the API stability. And that's where we really try to make sure that polymorph screening is performed even at early stage. And it should be robust enough, because it really tells us what is the most stable polymorph. And this stable polymorph only should be selected for the development. And most important, claiming different polymorphs in your patent gives you a possibility to have stronger IP portfolio. In the past, we must have heard, based on the salt screening or different polymorphs, maybe there is possibility to break few patents, and from that aspect also it's very important. Then salt screening is another aspect we need to consider, because if you see more than 70% of candidates in the development pipeline, they are either weak bases salts, and often you do have some issues with respect to the solubility or stability. Maybe often these weak bases or weak acids are not soluble enough. That does not allow you to have an oral formulation which is showing very good bioavailability. For the parentals, at least you need a formulation where 100% drug should be in the solution form. And that's where the salt screening is very, very important, because the salts are ideally highly soluble compared to the parent compound. The salt screening also allows us to expand the IP portfolio. But in that case, then again, careful consideration must be given what kind of counter ions we are selecting. You must know the issues with counters like mesylate, tosylate. If this is carcinogenic impact, it should be ruled out. Then particle size optimization, because the API particle size, it has a direct link with the dissolution and thereby the bioavailability. So the API of the pH of the API must be controlled. It should be monitored, because for the oral solid dosage form suspension, your API is always going to be the solid state. So if there's any change in the particle size, it will directly impact your bioavailability. For the parenteral suspensions, typically the intramuscular subcutaneous injection, again, particle size is very, very important, because if there is any change, it will have the different release profile, different exposure, your safety and efficacy profile will certainly change. And in that case, then often the pH is impacting stabilities. And this is typically true for hygroscopic APIs, because higher the surface area, then higher the moisture content or moisture uptake is there. From that aspect, also pH is very, very important. So typically during the last step synthesis, which is crystallization step, which we try to control very carefully, that so that we do have the reproducible particle size. Even particle size is the part of specifications, and we really check that obtained product is complying with respect to the specifications. If at all micronization is needed, then we try to do it using by jet milling, where API is subjected to the micronization, or in some cases, even the crystallization step is optimized in a way so that the micronized API is possible. Then we should also focus on the specified and non-specified impurities. There's a clear guidance from ICH Q3A, which really tells us what should be the limits for the different impurities. The typical impurities we do have, like organic impurities, inorganic impurities, and residual solvents. And what are the sources of these impurities? They might be coming from starting materials, byproducts, intermediate. It might be API degradation products, might be coming from reagents, ligand, or catalyst. So, and there is a clear guidance which is also linked to the dose of your compound. So if the dose is, let's say, higher than 2 grams, then you have different reporting thresholds, identification thresholds, and quantification thresholds. So when I say reporting threshold, this is mainly coming from your chromatographic purity. Then you need to report those impurities, that what is level of that impurity at the release, whether there is any increase or the stability storage. When I say identification threshold, so at least you should know what is the identity of that particular impurity. At least you should know what is source of that impurity. In terms of quantification, often if it is really, as you see the limits, if it is going above 0.15%, so in that case, it is very important that that particular impurity is also part of your tox studies. So when you do your tox studies, not only your compound is dosed, that particular impurity is also being dosed, and you already know at that particular concentration of that impurity, there was no adverse event. And this really allows you to set the specifications of your drug when you go for cleaning, because by that time you already know the effect of that particular impurity at that particular concentration. Then there are also limits on residual solvents, because we often use organic solvents for the API production, and this ICH Q3C R6 provides the guidance in order to check the level of these organic solvents, because of course these solvents come with some carcinogenic risk, and even this trace amount of these solvents, they have the impact on the physical as well as chemical stability. So when I say chemical stability, some solvents may lead to change into the solvents, the polymorphic form is changed. Some solvents may trigger the chemical degradation of API, and for that reason we need to follow the ICH limits for different solvents. On this slide, I try to put the typical limits for the solvent like acetonitrile. So what kind of typical exposure we might have, and that really tells you that what kind of concentration is allowed into your drug substance, and basically this is in ppm. So each batch is really checked for residual solvent by GC analysis to make sure that that particular solvent is not out of specifications. Then batch to batch consistency at the early stage is very, very important, because at the very early stage, we do have typical batch sizes 10 to 100 gram, maybe lower. But when we go for GLP talks or first in man studies, the typical batch sizes are even in the range of 1 to 5 kgs. So, and we do this tech transfer or scale ups we might have some differences with respect to the particle size, impurity profile, residual solvents, polymorphic form, even organoleptic properties. You may see some different colors. So this is very carefully monitored and controlled so that we do not want to have any batch to batch variation, because ultimately it is going to impact the performance of the drug product. So this was all about drug product. Let's try to discuss a little bit about drug. Whatever we discussed before, it was related to drug substance. Let's try to check what is the impact of physicochemical properties of the API on the formulation development. What sort of properties we should take into consideration. So PK is very, very important, as we discussed before, because the ionization of any API will tell you how this molecule is going to behave in vivo. Of course, we know that ionizable compounds are more soluble compared to non-ionizable compound. It also has impact on its passage across the cell layers, because typically the non-ionized part of the drug is going to cross the, let's say, the cell walls. Then LogP is also very important, because as we discussed before, extremely lipophilic compounds are difficult to formulate. So you need to have some balance. But same is the case for very hydrophilic compounds. I can give you example. We had one molecule into the pipeline which has a log-p value of -0.5. So in that case, it was so hydrophilic, by the oral route, the permeability or the absolute bioavailability of 0, then we had to develop the parental formation in that case. So that's the reason then you need to have some balance with the partition coefficient. But to give an example, when you are developing any anti-cancer drug, antiviral drug, the receptors, they really need some lipophilic properties of the candidates, and certainly you do have the compounds there on the upper range of the log P, but you may have some compounds who are working which are working onto the cell wall, some enzymes, and there may be hydrophilic properties are desired, and there you do have the compounds which are on the lower range of log P. And I said before, then log P, PK, they both really dictate the permeability, so permeability of the compound should be checked. Whether your compound is hygroscopic, because this will certainly have the impact on the formulation. Imagine you want to develop the oral solid dosage form like tablet or capsule. If your compound is so hygroscopic, so this will have some impact. Then flow properties, again, for the tablet or capsule formulation, flow properties certainly going to impact. Solubility is something very, very important. How your compound is soluble in the aqueous atmosphere, typically over the entire pH range, starting from stomach to the intestine, when you vary the pH between 1.2 to 6.5. And at that particular pH, if your compound is not soluble, you do have some issues with respect to the bioavailability. Again, what is the stability of your API against the pH? Imagine if your compound is highly not very stable or highly unstable in stomach pH, then in that case, you can't really go with the simple formulation. You might need some entry-coated formulation. Then what is the stability of solid and solution state? If your compound is not at all stable in the solution state, and if you have to develop IV formulation, you can't really go with the liquid formulation. You certainly need some lyophilized formulation in that case. And then what aspects we need to consider when we are trying to develop the preclinical or clinical formulations. So preclinical formulation, they are often administered by gavage. So formulation is naturally going to have suspension solution. Often I have debate with formulation scientists. They try to come up with the solid dispersion formulation for preclinical studies. But at the end of the day, if you want to administer that formulation by gavage, you are going to put your solid dispersion into the aqueous phase. All the amorphous compound will precipitate out. So what is the advantage of that formulation? So this is also one aspect we have to always consider. The poorly soluble compounds, they always present a great challenge for the dosing, because imagine for the tox studies, the doses you are using, they are almost tenfold higher than your clinical dose. So you really try to put a lot of drug into the animal species, because you want to achieve some MOEs, multiples of exposures, because you really want to find out at what dose there is no adverse effect. That's what we discussed before, NOEL. And for that NOEL, the dose used for the tox study is really high. And it's not so easy to formulate those higher amount of doses. So in that case, if your drug is very poorly soluble, it makes really life very complex. So in that case, you need to use the enabled formulations, which I will discuss in the next slide. So first in formulation, which is typically your phase 1 formulation, it has to be kept as simple as possible, because the aim is really to check how safely your drug is tolerated. Because imagine you spend a lot of time to develop the first in man formulation, but you find out the drug was not very well tolerated in the clinic. So all the efforts you have put on the formulation development, they are waste. So And that's the reason they ask us to keep it as simple as possible. I can give you examples. Sometimes even we have just kept the API into the capsule, that's it. And then we really try to check how the drug is being tolerated in humans. Sometimes we go for tablets, sometimes even you go with the two component systems. One is containing the API and other is containing the ready to use vehicle. They are simply mixed just before the administration, and these are typically first in formulation. So for the BCS class 1 compounds, we all know BCS classification, so I'll not go into the details. So for the BCS class 1 compounds, so these kind of formulations are very straightforward unless there is any stability issue. And if your compound is not stable, of course, we have to really use the functional excipient to stabilize that drug. But whatever choice of excipients we are doing there, we have to always consider the FDA IG database to make sure what type of excipients, what concentrations we are using. And then when you have the BCS class 2 drugs, certainly you need to consider enabled formulations, because at the end of the day, we want to increase the higher exposure in vivo. So what kind of formulation we can consider? You may go for cyclodextrin complexes, micro suspension, nano suspension. So when I say micro suspension, so typically your API is micronized. When I say nano suspension, so this is not possible in solid state. So you just disperse the API within the aqueous vehicle. You may go for the nano milling, you may go for the homogenization, and that's how you obtain the nano suspension. Then there is also possibility to use the lipid-based formulations like liposomes, microemulsions, mixed micelles, mesoporosilica particles, and solid dispersions. Also other options where you try to convert the crystalline form into the amorphous form. So I think this is a very broad discipline. So I mean, a separate presentation could be possible on this topic, but I just wanted to give a glimpse at what is to be, needed, or what is to be decided in case you have some bioavailability challenges in your preclinical phase or clinical phase. And of course, whatever decision you are taking at the end of the day, which technique, it is often driven by the budget, because a lot of finance needed. In this case, you need to outsource these trials to the specialized companies. And timelines, because these are not simple formulations, these are the complex formulations. So naturally, it is going to impact your timelines. And at the end of the day, what are the chances of success? Because you try to imagine something, okay, this approach is going to work, but before you have the PK data, you cannot guarantee that. So the chances of success also need to be considered. So with that, I would like to conclude my talk. So drug discovery is a vast project, as we discussed before, with the huge cost, times, and risk involved. So one should be aware with that. It really requires various disciplines, the departments, they are working together, maybe from pharmacology, toxicology, CMC aspects, pharmacovigilance, and they really try to address the challenges in order to make sure that we have the highest possible efficacy, the productivity is high, failure rate should be low. The advances in genomics, bioinformatics, they really help a lot to predict the probability of success in the drug discovery project. Also, it helps to reduce the overall cost. From the CMC point of view, we should really try to highlight what are the possible challenges, what are the possible risks, so that we can really give some orange or red flag to the team, so that the late stage failure can be avoided. So with this, I would like to conclude my talk. Thank you for listening, and sorry for the technical break we had.
Thank you. Again, I would like to invite our moderator, Mr. V.H. Jain sir, for session conclusion.
Once again, good afternoon to all. As a moderator, once again, I am here to conclude the session. Definitely, sir, whatever information you have been shared with our participants and the students is highly fruitful, and thus it was in the very learning and the talkative session. However, sir, you have been covered all the information and all the informative concepts related to the new drug discovery. Sir, you have been covered the topic of CMC challenges, which is a very important topic in the documentation section while filing for the IND and NDA. Sir, you have also explained nicely about the phases of creation of the new drug. You have explained nicely about the processing. And also with the typical screening process, you have been went through that particular side. You have been also explained very nicely about the recent topic of CAD, which is recently introduced by the PCI in our MPharm syllabus. So the session was highly helpful, informative for the researchers about the softwares and the bioinformatic database also. So once again, we thank you, sir. And sir, there are the few questionnaires. Now this session is going to be open for the questionnaires. So if the participants are having any kind of question, they can just put down that question into the chat box so that we can highlight that particular question. So this was in the first question. So question is from my side only. My question is that without CMC information, are we able to proceed for the generic drug application? No doubt for IND and NDA it is necessary, but for ANDA.
Yeah. So what do you mean by CMC information? So when I say CMC, it's a chemistry, manufacturing, and control. So when I say chemistry, you certainly need to know the structure of the molecule. So without having the structure of the molecule, you do not know what kind of synthesis you need. So that's
Fine.
where the chemistry is coming into the picture. Once you know the chemistry, then you go for manufacturing. So you certainly know what is the process we want to use to produce the drug substance, what kind of dosage form we are going to manufacture, with tablet, whether it is parenteral formulation, what kind of primary packaging you are going to use. And then comes the control. So what kind of control you have in terms of quality? You need to set some specifications for your drug substance, drug product. So you said whether it's a generic or new drug, doesn't matter, but CMC is always very critical. And this information is always expected in your NDA dossier as well as ANDA dossier.
Okay, sir. Fine. Thank you, sir. So this was then the second question. This question is from Himanshu Patil.
Yeah, I mean, are you going to read or I can answer directly?
Yeah, question is there, sir, on the screen. If you ask, I will read, sir. Why clinical trials require too much time? If any pandemic situation is arrived, then what about that medication required for that particular disease?
Yeah, so I mean, how I can start? I mean, very interesting question, but you have to also see the consequences. At the end of the day, I said the risk versus benefit. You want to develop a drug, not a poison. Okay. So, and for that reason, the safety aspects must be addressed very well. That's what I was trying to point out. That at the preclinical level, you have to check your drugs should not be carcinogenic, it should not be mutagenic, it should not be impacting the reproductive toxicity. Then you go for humans. Even into the humans, you first it is done on volunteers. If I ask you, okay, this is a new drug I have developed, would you like to inject? Of course you will not be ready, because you want to see that what is the dose, what are the adverse effects. And that's what we want to do in the phase 1. We really want to check whether the drug is safely tolerated, and that's where you try to check decide the dose of the drug for the next trials. Then phase 2, you try to check what is the efficacy of that particular drug against comparative. Imagine you develop a drug which is compared against acyclovir for COVID, and you find out that against acyclovir that drug is not active at all. So what's use of that drug? That tells you the efficacy. With that data, you go to phase 3, which is done even large number of patients, 2 to 3,000 patients. You really try to check how drug is getting absorbed, how it is being metabolized, whether any metabolite is being toxic. Because at the end of the day, again, I'm repeating, you don't want to develop any toxic drug, you don't want to have any adverse events. And that's the reason overall drug development really needs a lot of time. You can't really develop like this.
Okay, sir. Thank you, sir. So this was in the next question. This question is from Honorable Dr. Shirsagar. Drugs should not affect the important organ of body during the course of treatment of one disease. I mean to say that what is the your opinion, means opinion of Dr. Yogeshwar sir?
Yeah, sure. I mean, a very interesting question. Thank you, Sambhaji sir, for being there. I mean, as I said before, a lot of drugs are being eliminated by kidney, because kidney is a major, let's say, organ for metabolism. So naturally, the drug is being exposed to kidney in that case. So imagine you are developing a drug, it is binding to the particular receptors, but at the same time, when it is getting excreted through the kidney, and if it is causing any nephrotoxicity, certainly this is something you would like to avoid, and all these aspects you need to take into consideration. Imagine you are developing a drug which is taken by oral route, but if that drug is, let's say, some irritation potential into the stomach, into the intestine, and it may lead to some cancerous type of adverse event. So all these aspects also you need to consider, that when drug is being administered, through which organs it is being circulated, and whether we have any non-specific binding. And this non-specific binding should be avoided when we try to do these docking studies, where you really try to make sure your drug is really being specific for that particular receptor which is present on that particular organ.
Fine, fine. Okay, sir. So this was the next question from Honorable Dr. Sachin Sakat, sir. When the formulation development, means pilot plant development of the molecule, comes into picture in the drug discovery process, then sir's question is that it means it before or after the clinical investigation?
Thank you, Sachin, for being there again. So I will say it mainly comes at GLP tox studies, but typical formulation you need for GLP tox studies really more or less in the same range what you need for the phase 1. So I will say even at GLP tox studies, you really need a robust formulation. Again, there you want to make sure that your formulation should not be a showstopper. It should not be, it should not lead to some delay in timelines. So you really want to make sure that at GLP tox stage, your formulation should be upscalable. It should be stable enough, which is fit for clinic. And most important aspect, the focus is always to keep the same formulation for GLP tox in first in man. Otherwise, then of course there's an impact on the exposure. FDA may ask you for bridging studies, and this is something you would like to avoid.
Okay. Thank you, sir. So there is one more question from Himanshu Patil, which I am displaying on the screen. During treatment of one disease like diabetes, it's chronic disorder, so there may be chances to affect the organs like kidney due to the prolonged treatment. So what is your opinion? He was asking about your opinion, sir, on this particular question.
I mean, I may have to read this question again.
Okay, just a minute, sir. During treatment of one disease like diabetes, it's a chronic disorder, so there may be chances to affect the organs like kidney due to the prolonged treatment. That was the opinion of our participant, and he was asking about your opinion on this situation.
Right. So I mean, this is not from the let's say from the disease itself. It's also it's mainly related to the chronic administration. And I'm repeating again. Typically, it's also case with NSAIDs. Most of the NSAIDs, they are being metabolized by kidney. They are being excreted from the kidney, and that's where the kidney toxicity comes into the picture. So, and again, we are coming back to phase 1, where we really try to understand the ADME properties of your drug molecule, how it is being absorbed, how it is being metabolized, where it is exactly excreted. And that when this excretion is happening, we have to really focus what is the impact of the chronic administration of that particular API, whether it has any impact also on the metabolizing enzyme. So there are two aspects. One, first is what body is doing to that drug. Second is also what drug is doing to the body, not only to the target organ, but what about other organs? And this is something to be carefully checked.
Dr. Shiraz Sheikh, is there any effect of a drug on immunity is checked during the time of clinical trial?
Not necessarily, it depends on the indication as well. But yeah, I mean, it's always the case that when you are administering any drug, you want to check the adverse event. And if there's any adverse event, you also try to understand what is the mechanism for that adverse event. Imagine you do have some anaphylactic reaction, hypersensitive type of reaction. In that case, you'll certainly check whether there are cytokines released, whether there is any role of interleukins, but not necessarily always. But if you are developing any drugs for immunological conditions, imagine you are developing an immunomodulator, imagine you are developing any antiviral drug which is acting by impacting with your innate immunity. In that case, certainly you need to check the effect on the immunity, but not always. I will not generalize it.
Okay, sir. So sorry to say, but two more questions are there. One is from
No problem.
Okay, one is from Honorable Dr. Sandeep Pathare, sir. Here the question is on a screen. How many and what stages should drug developers and the discoverers plan for the bioactivity test of a drug molecule?
I mean, not sure if I really understood this question, but what happens, as we discussed, you try to first select the target, you validate that target, and then you go for in vivo assays. It may be antibacterial assay, it may be anti-cancer assay, it may be antiviral assay. So in the animal models, you really try to check if your drug is really working against that particular disease. If it is bacterial infection, you will try to induce the bacterial infections into the mice model. Let's start with the mice, and you really try to check if your drug is acting in that particular model, and you try to really correlate whatever in vitro data you had from the IC90, IC50 values, how it is really fitting into the in vivo data into the mice. Then when you go to the clinic, of course, in the clinic, you try to set some endpoints. Endpoints could be then what is the reduction in the, let's say, viral shedding rate. Let's try to put that. Or maybe it is a cancerous disease. You really try to check what is the decrease into the mass of the tumor. Let's try to put like that. And this type of how you will phrase pharmacological response or bioactivity test, you really try to gather the data to make sure that your drug is really acting as per the predicted mechanism, and if the data is really fitting into the in vitro data you have generated. So I will not say there are any standard set of bioactivity tests, or no one really tell you, okay, you do this kind of set. At the end of the day, you have to really make sure that you feel confident with your potency results, with your pharmacological response, and data is really appropriate for the submission in order to ensure FDA that your drug is safe and effective.
Okay. Thank you, sir. And the last question, that is from one of our alumni, Mr. Vijay Kuar, sir. When we use new excipients or existing one for the different routes other than those approved in IIG, then is toxicology data of the same with formulation will be enough or need to generate data separate?
Okay, thanks, Vijay. I think this is also a very important question. If you want to develop a new excipient, I will say it's similar developing a new drug. So you need to really generate all the data you need for the new drug whenever you are developing a novel excipient. If you want to really go into the details, you can refer to Captisol, which is a sulfobutyl ethyl cyclodextrin developed by Sidex and Pfizer in collaboration. So it really took a lot of money, lot of time to develop that new excipients. Okay, regarding the approved excipients, if you are really crossing the IIG limits, so naturally you need to prove that at that particular concentration, whether your excipient is safe. And for that reason, you have to generate enough toxicity data for that particular excipients. And then more or less, you are taking the same path which you take for your drug molecule. So I'll repeat that if any new excipients or let's say not aligning to IIG limits is as good as developing a new drug, and ideally no one will take that path.
Okay. Thank you, sir. Thank you very much. This is the end of questionnaire session. We are once again thankful to you, sir. But a few comments which I want to share here, which I received in the live chat box. It's really happy to hear such an informative session of Honorable Dr. Yogeshwar Bachau, sir. The participants has commented very informative session. It was an excellent session, motivative session. It was in the career uplifting session for the students, and also there is a huge demand of same. There is a huge demand of your presentation from our participants, sir. Again, our
Yeah, so as
participants are,
Dr. Poshitiva said, even my presentation has some confidential information. So we can discuss offline. I can take out some information, then it can be shared.
yeah, sir. Yes. And sir, our participants are also thanking you for answering their questions. So once again,
No
thank you,
problem.
sir. And I'm handovering this particular mic toward Mr. Amit Dhankani, sir, for vote of thanks.
As a coordinator, on behalf of organizing committee, I would like to thank the eminent speakers who gave their precious time for this webinar. I would like to thank the management of Pujya Sani Guruji Vidya Prasarak Mandal who gave us permission to organize this event. I would like to thank our principal Dr. S.P. Pawar sir. Without his continuous encouragement and support, this event would not be possible. I would like to thank all my colleagues for their help and cooperation. And last but not the least, from the bottom of my heart, I would like to thank all the participants from different parts of the country without whom the event would not become into a reality. Thank you very much. With this, I hereby declare the end of this webinar. Thank you very much.
Thank you. Bye.